हेलो। Ma'am, can you hear me? Hello? Hello?
Hello.
Yeah, can you hear me?
Yes, ma'am, we can hear you.
Now, just see, Naren should have also joined, Dr. Chirmule, so that the, the session can start whenever you like. Just see
Ma'am,
chirmule@gmail.com. Can you make him enter, please?
Is he joining from Symphony Tech Biologics?
Yes. Please.
So we have to share your slides from here itself, I think.
Yes.
Yeah. Okay.
a very good morning to all the dignitaries and distinguished guests. Firstly, I request the students who are sitting at the back, please come in the front seats.
Guljeet, why don't you start and then I'll take over?
No, Naren, this is fine. Okay.
So we are starting our 7th scientific session, and for which I would like to invite the chairpersons of the session, Professor Dr. Mridula Saxena ma'am and Professor Dr. Shishir Nandi sir. Mridula Saxena ma'am is the ex-professor of Department of Chemistry at Amity University and Honorable Trustee of Global Institute of Pharmaceutical Education and Research, Kashipur. Professor Dr. Shishir Nandi, he is the professor and HOD of Global Institute of Pharmaceutical Education and Research, Kashipur, and also the organizing secretary of the symposium. I request ma'am and sir, please join us on the dais. The speakers of this session are Ms. Guljeet Chaudhary and Dr. Chirmule, Professor Disha Datta, Professor Dr. Gunda George. And I request the students to pay attention towards the lecture.
They are from electric. Okay. Their area of work is gene therapy. Okay. A very good morning to all of you. I am very much surprised to chair this session, and I would like to introduce Mr. Gurjeet Chaudhary and Dr. Chirmule. They are working in collectics in area of collectics in. The area of their work is gene therapy, and they have experience of all more than 40 years. So I would like to invite both of them to give their presentation.
We would really like to thank Professor Sisir Nandi, and I understand that he also had a family issue yesterday, but he has still sort of at short notice given us this wonderful opportunity. Both Dr. Naren Chirmule and I have been in industry for over 35 years, and we have been in pharma industry leading in various companies. So just to actually, and we are very pleased, ma'am, that you have introduced, and I know that it was a second before, so I apologize for that. So coming to the point, my background is really for pharma industry, leading in Ranbaxy as a regional director, several international markets, introduced the generics. Thereafter, at Jubilant Biosciences and Reliance Life Sciences, the services part of the business, which was really building molecules and etc., which is drug development at Jubilant, and then clinical trials. Four of the major CROs which now contribute to over 5,000 crores in the country, which is Sinuous, as well as and in Sinuous, 4 CROs we brought in from the year 2005 to almost 2015. And these were United Health Groups, which is a very large global provider in the US for clinical research. So from 2005, and these 4 CROs actually combined together to what you see as Sinuous. In addition, PRA, which became ICON. So, and I have serviced over 17 of the top leading 40 pharma companies of the world, including Thermo Fisher as scientific advisor. Both our interests, Naren has devoted his life to gene therapy, and he will talk about it shortly. His CV is there. And we have been the early birds in introducing Immunil in Bangalore, working very closely in bringing the 2 major players in India today who have been approved by the drug controller are Immunact and Immunil. Immunact was the open system, and Immunil is actually now providing commercialized CAR T cell therapy. And we could talk for it for long, but I would like to capture Naren's view because he is actually on a flight to Bangalore, but he's very much on the call. So Naren, would you start, please? And we are very, very, excited. We would have been next time. We are definitely going to come to Tiger Resort. So he's also a very avid biker and Himalayan climber. Thank you, ma'am, and we'll see you probably at Amity. Yeah, we've all spoken at Amity several times. So go ahead, please.
Yeah. Thank you very much. And sorry about this last minute presentation that I'm doing from the car. And maybe you can see my video. I'm going to put it off, but I'm just going to say hello to everybody in the audience, just so that you know who I am. So let me take the next 5-10 minutes to give you an overview of my view of advanced cell therapy. So if someone can forward the slides for me. Next slide. So as Guljeet mentioned, there are several CAR T cell therapies and gene therapy in India, and it's a very, very exciting time to be in this field in India because we have an opportunity to do transformative medicine. The next slide. So what I mean by advanced therapies is listed on these slides. Predominantly gene therapy, CAR T cell therapy, inducible pluripotent stem cell therapy, and gene therapy vectors and gene editing. And there are advances being made in all areas in India. The next slide. This is my brief CV. I'm an immunologist. I have about 40 years of experience in developing vaccines and biologics in the United States and India. And I play the flute. Normally, if I'm there, I would have actually played the flute, but maybe that's another day. Next slide, please. So let me tell you a story about gene therapy in the next 10 minutes or so. Starting in 2015, this is 10 years ago. 10 years ago, the holy grail of gene therapy was these points that are listed on this slide: stable gene therapy, long-term expression, gene editing, targeting the gene insertion, gene replacement, regulation of gene expression, and prenatal therapy. And if you do one click on the slide, please. One click on the slide, yeah. And 10 years later, all of these have been addressed. All of these, what were very, very significant challenges, have been addressed. So today, with the advent of AI and ML, yes,
can I request just to go back, because there are students as well, to the previous. So if you can explain the right side of this column. It's very important because, you know, during this period, the Obama Act of you know, this is not a medicine. This is an actual precision therapy. So
yeah, I'm going to be talking about this in the next few slides. This is just an introduction.
Just explain the, you know, the products.
Yeah, I will. It's coming up in the next few slides. The next slide, please. So in the advent of AI and ML, and the advanced therapies and quantum computing, and not listed on this slide, is 5G and 6G communication processes, the ability to make medicines is going to be transformed. Next slide, please. And what better place to do that than in India? So let's talk a little bit about gene therapy. Next slide. So the story for gene therapy starts with my friend, Murli, who's today 64 years old and lives in Chicago. He has a complete mutation in a gene called factor 8. The absence of this gene doesn't allow his blood to clot at all. So if he gets a cut, he might bleed to death. If he climbs a flight of stairs, he will die. In order for him to survive, he needs to take factor 8 protein every day of his life for the last 64 years. If we have gene therapy where there's a gene that expresses the protein, he will be cured. So that's my story, personal story, and I talk to Murli very regularly and understand his plight and the challenges he's facing. Next slide, please. This is a short video. Can you click on this slide and please play the video? Oh, maybe in the PDF it is not playable. Sorry about that. It was, but let me just tell you about this lady. Her name is Sanjana Goyal, and she has muscular dystrophy. You can see that she's sitting in a wheelchair. Her legs are strapped, and you can see her hands. With the mutation that she has in the gene, she is unable to lift her hand or move her legs on her own. She has to be fed in the mouth, and every action that she takes has to be assisted by someone or the other. She lives in Solan, and she has built an institute for therapy of muscular dystrophy patients. Working with patients like Murli and Sanjana is critically important to be able to understand how the impact of gene therapies that we are working on patient lives. So next slide, please. So what is gene therapy? Gene therapy the story of gene therapy starts in 1972 when the first recombinant DNA technology was invented, and Genentech, the company in San Francisco, was formed. Over the next few decades, several major events have taken place, including the human genome being sequenced in 2000, and finally the approval of the first gene therapy drug, Luxturna, for treatment of blindness in the eye have been reported. I've written a book called Good Genes Gone Bad that you can see here, which is available on Amazon, which I've tried to explain the entire story of gene therapy in layman terms. So I would encourage you to read this and other books about gene therapy. So let's talk about 2025. Next slide, please. One more click, please. Yeah. So the first step in gene therapy is to identify the target. And today, with omics technology and the ability to analyze data, finding a needle in a haystack has become much easier. So we have 21 trillion cells in our body. Each cell has 3 billion nucleotides. These nucleotides express at least 30 to 60,000 genes, proteins in each cell. So the amount of data that we can get from every single individual is very huge. And it is the process of big data analysis that will allow us to find genes which are responsible for genetic disease. This is an exploding field. So for students in the audience, this is an extremely exciting time to be doing research. Next slide, please. In order to transfer the gene from outside the cell to inside the cell, you need something which is called a vector. One of the major advances in the field is the identification of this virus called the adeno-associated virus, which is an excellent vector for transducing genes into the patients. And these three scientists, Arun Srivastav, Jim Wilson, and Jude Samalsky, have made seminal discoveries and inventions to be able to harness the power of adenoviruses to become gene therapy vectors. I'm just going through this slide deck fast, but I encourage the student to take notes and do some background research on the topics that they are hearing. Next slide. What Arun, Jim, and Jude did was, and of course many other scientists in the field, identified many different AAV vectors which are transducible to many different organs. Next slide. Gene Bennett was one such scientist who, in University of Pennsylvania, who utilized AAV vectors to express a gene which is mutated, which is called the retinitis pigmentosa esterase 65 gene. It's a 65 kilodalton protein. When the gene for this is mutated, children become blind. What she did is she expressed this protein in an adeno-associated viral vector, injected into the eyes of blind children, and now the children can see. Next slide. Yeah. Spark Therapeutics, which is now purchased by Novartis, developed this technology. It has taken more than 30 years of Gene Bennett's work to come to this stage to get this eye gene therapy approved. I'm not going to read the details of the slide, but I encourage you to read the clinical trials for Luxturna to understand how this therapy was proven to be successful. Next slide. Similarly, Kathy Hai is working on developing a gene therapy using AAV vectors for hemophilia, and we are very close to developing this gene therapy. The next slide, please. There are many companies who are doing clinical trials with this vector, and within the next year, maybe hopefully in 2025, we shall have approval for factor VIII, which will enable Murli, my friend, to get the treatment and hopefully help him live his life, live a better life. Next slide. So that was AAV gene therapy. Now, quickly moving on to CAR T-cell therapy, we don't have time to go over the details, but maybe in your session, in your conference, you'll hear much more about this. Carl June, Mike Sutherland, Stephen Rosenberg have done extraordinary work in creating this immune cell, which is now curing cancer. And there are 4 books that are listed here: Breakthrough, Song of Cell, The Cure Within, and Living with Cancer, which I strongly encourage you to read if you're interested in this topic. Next slide. Next slide. And finally, we come to gene editing. And Church, Doudna, Zhang, and Carpentier, and Doudna and Carpentier won the Nobel Prize for this discovery or invention, where they can take a gene, edit it, and correct the gene defect. This again is written in a beautiful book called The Code Breaker. I encourage you to read this book. Next slide, please. And finally, just a summary of the kind of things that people are doing in the field of gene therapy: AAVs for treatment of muscular dystrophin, mesenchymal cells for ALS, gene editing for sickle cell anemia, and antisense oligo for ultra-rare diseases of single-point mutation genetic diseases. I think this may be my last slide. The next slide. Yeah. So why don't I stop here? I can tell you a detailed story about muscular dystrophy, but I think maybe running out of time. So why don't I stop here and see if there are any questions? Thank you very much.
I would just like to add here now that there are about 15 to 20 companies working in India, including the large players like Dr. Reddy's and, have apart from, and the hospitals as well. And the government, there has been a recent last month STEM meeting under the Prime Minister's Committee, where a lot of incentives are being given to startups. And BIRAC is very highly engaged. Actually, Naren is just walking into that next meeting for BIRAC on the committee, that cell and gene therapy is highly encouraged. In case any of you would like to know about the clinical trials also, for which we are highly equipped now in India, we could be supporting you in terms of your questions, answers, and your project on a pro bono basis in terms of knowledge. We have a group which is of cell and gene therapy, where these lectures are done, periodically, and sharing our upgrades, as it were. But this has meant a lot of, collective work. I'll just give you an example that before this, 21st Centuries Act was done in the US, and during the Obama administration, Naren will tell you that there was such a hard stop for 19 years because of the death of the first gene therapy, until it was the first product to be approved. Naren, can we go back to that slide of the products which are approved in the world and are available?
Yeah. I don't have control. Somebody
has. So, sir, could you go back to that list, because you should know what is available in the world, and every country. And you'd be surprised, more than Europe, it was US and China. China was far ahead of us, and they were the second largest after Europe in 2022 as well. We go back, there's another one which was much earlier. That's a third slide, please. And we would encourage any questions that you have. It's a large topic to cover. It was just to stimulate your interest in this area, since many of you may have a medicine background and a chemistry background and so on, biochemistry as well. This one, yeah. So Kymriah was the first one, which is with Novartis. Carl June is the inventor, and Carl June is sitting on the boards of 2 companies in India as well, both Immunact and Immunil. So that's the kind of support we're getting, but we were nowhere on the global map earlier. We have just come in 2024, basically on the global map in terms of having a commercial product. Then there's for gene editing, Cascavy, etc. Now, Naren, can you mention a few more if you'd like, which are already approved? Huge number are approved by BMS and many global groups.
Today, there are 6 CAR T-cell therapies that are approved, all for different cancer indications. And there are at least 2 or 3 gene editing in very late stage clinical trials for sickle cell anemia, for thalassemia, and also muscular dystrophy. Yeah.
And finally, it is now in our own country, the PM has really encouraged affordability and access. We also have gene therapy works much more as far as immunology and where we are hopeless situations and rare diseases and orphan diseases. And we have the legal ability today where there is a judgment that even up to $50,000, those who are approved by AIMS can get lifetime support. So things are changing very rapidly for the better. And there's a huge canvas of play that is happening right now. As I mentioned to you, there are startup and camps all over the country. Every state is encouraging, but really the very prominent ones are Hyderabad and the locations around Hyderabad, Bangalore.
Sorry to
Bombay.
interrupt you, Guljit. I have to log off, but if there are any questions, I'll be happy to
Please
answer. Then I have
ask
to log off.
questions. Yeah, and our emails are given below. But please, any student, ma'am, you, anybody else has, sir, any question, please let us know. There's a lot of training that has to go back into the ecosystem, both in terms of clinical trials, the regulatory aspect. Drug controller has launched a separate the guidelines have been put in place for gene therapy, and there is a 15-year follow-up each of the patients. As I understand today, there's a 5,000 queue for oncology treatment between the two players who are approved. So any other question? This is all done in BMT, so there's an infrastructure issue also because it's only in bone marrow transplant that you are able to actually do the patient outcomes. And
Hello.
Yeah, go ahead, please.
Yeah, I think very nice lecture and information, I would say, and to all the audience in this. I would just like to know that you are right in this slide. It is holy grail for gene therapy. Now, these are when you are writing in clinical trials gene replacement. So it will be for certainly gene replacement for hemophilia for that particular disease, or it is in general. What does it mean? Regulated gene expression preclinical. So it's a name.
Yeah,
normally, you put the name of a particular product into that.
Yeah. So on the left side are the challenges that were listed in 2015, 10 years ago. So you asked for gene replacement therapy, right? So, for example, now, if you have a thalassemia, if you have a sickle cell disease where the hemoglobin gene is mutated with a single point mutation and doesn't result in development of the hemoglobin, the technology that is available with gene editing today is we can cut the gene in the allele off and replace it with the normal gene. So that's replacement gene therapy.
Okay. So, so it's a kind of a general thing, or it is for a particular specific which you are talking about?
Right now, the technology is available for a single gene mutation, but it won't be far before we can delete many genes and replace them.
Okay. And I think there is a therapy which is quite expensive for SMLE disease, which is in children, but the cost is very high of that. So how are you thinking of cutting the cost, in particularly in gene therapy?
Well,
I think this is a very major issue.
Very big, but yeah, the cost of these drugs, whether it's gene therapy or any other drug, requires another discussion that we can have another day. But in a very short summary, I would say, I know more about the United States than other parts of the country, other parts of the world. Pricing in the United States is not based on the cost of goods. It is based on what value it provides. I'll just give you an example. For factor 8, my friend Murli, he takes recombinant factor 8. And for every injection that he takes every day, it costs him $4,500 per day. So you can calculate it's about $800,000 a year. And if you calculate what that is, about $1 million a year, which is 7 crore per year he's paying for the last 40 years. Nobody can afford this, right?
Yeah.
But the insurance is paying. So why does it cost 7 crore for a protein that probably costs, I would just give $10,000 to make? That is because the value is based on how much money are you willing to give the system, whether it's the industry or whatever, who the receivers are, for Murli to survive in 1 year. How much value is Murli for? His value is 7 crore they've established. If his value is 7 crore, then you must pay 7 crore. And that's how pricing is done. In India, I think we have a great opportunity to change the way, at least for these life-saving therapies, to price it based on cost of goods. So we can bring that cost of hemophilia from 7 crore to maybe 70,000 rupees.
Sir, scale up is extremely important on the one side, and scale up is very different. You don't have a pharmaceutical factory here because you do have there are two kinds. One is the in situ setup, which is per patient, like a Miltony Prodigy individual. You are taking the cells from the patient and then reinserting them back after a certain period of time, having done all the apheresis. The other is the open system, which the large players like Novartis and all have, and which actually is real scale up. Now in India, the real scale up is just
sorry, Guljeet, I have to log off now.
Yeah, sure.
So
sure,
thank you very much for the opportunity, and hopefully I'll connect with other people again later. If you want to reach out to me, you can connect me on LinkedIn. Thank you. Bye.
Yeah,
thank you for the opportunity.
Your mobile number as well.
Yeah, no, not mobile number, but
not mobile,
LinkedIn is
email.
better.
No, email.
LinkedIn is the best. Thank you so much.
Hello.
Okay.
Guljeet.
Yeah. Yes, sir.
Thank you and thanks, Dr. Narendra, for presentation on Holy Grail Gene Therapy. My name is Dr. Shishin Nandi. So I am organizing of this symposium, and thank you very much for the support. I have one small doubt. If I ask you, the genes which are responsible for aging, in our normal life, is it possible to replace that gene and incorporate the new one to make young the people that they will never be going to aging?
So Shishir, one thing I will tell you,
Sir, please.
Technology is not the technology is not the limitation anymore. Your imagination is the limitation. If you can imagine it, it can be done.
Is it practically possible? Yeah.
It is practically possible today.
We have to go. This is absolutely true. That depends on certain time, 80 years, 90 years, 100 years.
Yes.
But is it possible that the people will go by young only without having any, that aging?
If you can imagine it, it will happen.
Imagination only. But now practically, is it that research is going on that on aging,
yes.
how to combat the aging?
Yes. There are a lot of research. In the United States, there's an entire National Institute of Aging that does. There are 500 scientists who only work on aging.
Okay, sir. Nice. And one small doubt is there. The people, particularly young girls, they are very crazy to go to the beauty parlor, to increase their beauty of hair and face like that. So is there any that, that dark complexation on the face and change that gene which are responsible for dark complexation and to enhance the beauty? Is there any clinical trial or treatment is going on that?
Well,
that
I don't know about.
Also. It would be non-toxic. It would be non-toxic treatment.
I don't know about clinical trials, but Shishir, again, if you can imagine it, it will be done. Technology is not the limitation. If you can think that if you can find the gene that is responsible for, let's say, changing the color of your skin from brown to beige. If you know the gene, and it's probably multiple genes, if you know the gene, you can edit the gene. There's a whole different question of whether that is ethical or whether it is allowed by society, and that is a whole different debate. But as far as the technology is concerned, the technology is available to do it. So I'm only focusing on the science part of it. And I think it's important to think about those, what are all the questions that you're asking, but there are 10,000 extremely rare diseases which are desperately need for gene therapy. I think those are the ones we should address first.
Okay, thank you.
And that's the health economics of the country. That's how, even in the US, it's a different thing for value, but in India also, one has to justify who you would be supporting under insurance. But cell and gene therapy now is being taken under specific kinds of insurance. I would like to thank you, Professor Sisir Nandi, and for Indira to connect us as well, Professor Indira Ghosh. Thank you, ma'am. May we log off?
Now the next speaker is Dr. Disha Datta. She is an accomplished academician, over 17 years of experience in pharmaceutical education and research, currently serving as a professor at Devasthali Vidyapeeth College of Pharmacy, Rudrapur, Uttarakhand. She has mentored so many students of M.Pharm and specialization in pharmaceutics. Her area of interest is cosmetology and novel drug delivery systems. She has delivered guest lectures, including a DBT-associated talk on COVID-19 treatment, and at a session at the Sage University of Biosensors in Disease Diagnosis. Dr. Datta has authored a book on pharmaceutical engineering and CAD and contributed to the book of chapter on microbes in baking industries. So I invite Dr. Datta to give her presentation. Thank you.
Thank you very much, ma'am, for the kind introduction. Good morning, everyone present here. I'm truly honored and privileged to be here today and stand before esteemed speakers, researchers, and distinguished guests who have contributed in the field of pharmaceutical sciences. And before I start my presentation, I would like to convey my gratitude to respected Dr. Saxena and Dr. Teotia sir and Dr. Sishin Nandi, who is always a guiding light for me, and the whole organizing committee. And as my topic for presentation today is development and assessment of cubosomal nano formulation loaded with beta cytosterol for improvement of antioxidant activity. This work I have done in the Department of Pharmaceutics and in Devasthali Vidyapeeth College of Pharmacy at Rudrapur with my some co-researchers. Here we have prepared cubosome, as it is one of the novel and latest I would like to say the particle of the zomes family. We have heard about these nanosomes or herbosomes, liposomes, but the cubosomes are the most stable particles, and having nanostructured lipid bilayer structure of lipid and water with two continuous water layers within it. And the cubic, it is a three-dimensional cubic structure, colorful structure, which we can see on the slide. The yellow part is showing the lipid part, whereas the bluish part shows the water bilayer, which is present inside the cubical structure. So why we have selected this cubosomes, as I have said, that it is the most stable in the zomes family and easy to formulate in the premises or the facilities which we are having in our institute. These cubosomes are providing controlled and sustained drug release, reducing dosing frequency, improve patient compliance, and minimizing toxicity and side effects. They also have improved penetration through the skin because they have dual pathways, both for lipid carriers, lipid soluble drugs, as well as the hydrophilic drugs also. They produce an occlusive effect over the skin, so that the drug absorption is easy through the skin, as well as reduce in water loss. And then they are made up of biocompatible polymers, therefore non-irritating and non-toxic to the skin. These are next is the targeted and localized drug delivery they provide, and thus the nanoparticles ideal for treating dermatological disorders. we are planning to make some cosmetic formulations in future using this cubosomes as a basic structure. So the drug which we have selected is the beta-sitosterol. Beta-sitosterol is a plant-derived phytosterol. It is present in many of the fruits and plant-based materials, and they have the property of hair health as well as skin health. Then also have shown in some tissue repairment, then activity in tissue repairment, and many studies which I have mentioned in the next slide, please. In this slide, that beta-sitosterol is having anti-diabetic effect also. But here what we are trying to make it an anti-aging emul gel or a gel using these cubosomes. So why we have selected beta-sitosterol? Because these are having very less or negligible solubility in water, and these cubosomes can take up this beta-sitosterol because having this lipid environment, and because of their less solubility, they are also have less means very prone to oxidation. So to prevent those oxidation also, we want to introduce it in the cubosomal formulation. So the rationale of work we have decided is the enhanced solubility and bioavailability of the drug, so that we can increase the availability of beta-sitosterol to the skin or for hair health, then sustain and control drug delivery, so that there is no need of frequent dosing, then increase skin penetration through topical application, because I have said that cubosomes forms a occlusive layer on the skin, and because of this lipidic property, the drug can easily penetrate through the skin. Then high stability and protection from degradation. Because beta-sitosterol is prone to oxidation and degradation, cubosomes provides a protective lipid matrix, increasing the shelf life and stability of the drug. Then biocompatibility, both the things are derived from plant. So the polymer which we are using for the cubosome preparation is glyceryl monooleate. It is also a plant-based lipid, and polaxamer 407, it is used as a stabilizer here, and with with the help of water, we have prepared this cubosomes. Cubosomes are having highly crystalline cubic structure, and they are can be prepared with 4 different methods: top-down method, bottom method, emulsification, and spray drying method. But here we have used emulsification method because it was comparatively easy to prepare, and the particles which we receive from this, they are quite stable and can easily penetrate through the skin. This is next slide, please. So this was the rationale of work which I have discussed. Next. Then cubosome preparation. Here 4 different methods which I have told. Top-down method, it needs high energy, shear force. Then next is the bottom-up method. Here the cubosomes which are made up of due to through bottom-up method are quite unstable, and then next is emulsification method, and last one is spray drying method. So we have opted the emulsification method. Next slide. Next slide. So here is the step which we have gone through. These are the real images, pictures which I have taken from in the laboratory. Here, the first step is selection of components. We have used GMO, that is glyceryl monooleate, and polaxamer 104, and the drug, because it is an oil soluble drug, and aqueous phase. We have taken both the phases in 2 different beakers. In the first step, we melted the lipid phase and introduced the drug in it, and then in the second beaker there is water in which we have dissolved the polaxamer and polyvinyl alcohol. at 70 degrees, and then added the second step. We can see the lipid phase is added slowly in the aqueous phase, and stirring with continuous stirring, and emulsification with ultrasonication is followed, and finally self-assembly of cubosome formations takes place with this. Next, then next, these are some of the calibration curves of beta-cetosterol, which we have taken in different pH and buffer solutions, as well as methanol, because we just want to see in which the graph is means the drug is more stable. Then we use the 5.5 pH for further study, because it is useful for our topical formulation. Next. These are the differential scanning calorimetry we have done for the compatibility study of the drug and the other components which we have used in the formulation. Next. And this is the formula which we have used for the formulation. We have prepared 9 different formulations and in different ratios of polaxamer, polyvinyl alcohol, and drug is taken 100 mg in all of them. Next. And these are some of the characterization studies which we have done, like clarity, pH, particle size, polydispersibility index, and zeta potential. In vitro studies, drug content, entrapment, and antioxidant activity, which were feasible in our lab. And next. So after doing this drug content and entrapment efficiency, we have found that the F2 formulation was found to be have most drug content, released drug content, and entrapment efficiency. Next slide. So we have done the further studies with the F2 formulation, and here we have determined the particle size of the F2 formulation. This came to be 139.7 nm, and the polydispersibility index is 0.25 of the optimized formulation. Next. We have also gone for zeta potential analysis, because we have not separated the cubosomes or isolated it from the emulsion which we have received. So we want to see what is the zeta or the characteristic of the particles within the colloidal system. So here the zeta potential came near about minus 15.17 MV. Next slide. Here is a SEM of the cubosomal formulation, which are showing some cubosomal structures. We don't still have the reports of TEM, so I have not shown it. Next slide. These are the in vitro release which we have done in the lab. And this is the graph of in vitro release. Here we can see the red square graph, which shows the in vitro release study. It has done for both beta cetosterol drug as well as the loaded cubosomes over 24 hours, and using phosphate buffer pH 5.5 as the release medium, because we want to make it for the topical formulation. The result for the optimized formulation indicated that initial 2 hours the drug release was 27.56%, whereas in after next 20 hours it was about total 92.44% drug was released, and cubosomal formulation displays a rapid initial release in the first 2 hours, followed by a gradual release throughout the remaining time. So next, we have also gone for these calculations for this release pattern, zeta zero order, first order, Higuchi, Cosmier, Prepaz. Next, these are the graphs for that. And here, after the study, we have seen that the maximum the release kinetics have shown that The first order kinetics as well as Higuchi model, our formulation is following for the drug release, because it is almost near to 1, near about 1. So we can say that initial stage, our formulation follows the first order kinetics, and in the second, after 2 hours, it follows the Higuchi model, where diffusion works for the release of the drug. Next. And now this is the antioxidant activity which we have done by the DPPH method. DPPH is a purple colored solution. When we put the drug in it, the color of the solution goes to pale or light colored, which can be observed in 570 nanometers. So then we have observed it in for different concentrations of the formulation, and we have also calculated the IC50 values for the same. Here is the graph for the DPPH activity. So next, so the conclusion in the present investigation is we have attempted to prepare a cubosomal formulation with with the intention of preparing a topical final maybe emulgel or maybe some cream to this formulation in future. We are also undergoing this stability studies, but I have not reported here because the study is still going on, and we are also planning for the animal study further. And next, these are the next slide. These are the references, some of the references which I have used, and this is my institute where I have done the my work, Devasthali Vidyapit College, and these are my co-researchers. The corner one is one of my MPharm student working with me, and the two on the right side are my colleagues in the pharmaceutics department. Thank you. Thank you. Thank you, ma'am.
The drug delivery system is very important in treatment of different diseases.
Yes,
and the targeted delivery is also important to reduce the side effects
yes,
due to systematic circulation. So you have highlighted the different techniques. Thanks for the presentation.
Thank you very much.
It's open for discussion if you have any question.
Please, सामने आओ.
So, thank you for the presentation. And what I would be a little bit interested in, we know that we have drugs which we deliver through IV because of the bioavailability being an issue. And then specifically when we are working in field of cancer, in specific chemotherapies, of course, few are coming as oral, most of them are IV. What do you think that this cubosome would have an advantage, and potentially it might help in converting some IV preparations go for an oral one?
Definitely, sir, it will work. I have gone through some research papers also, where some of the researchers are doing the studies in combating this diabetes mellitus also, and some cancerous cells also, because the drug loading is very good in this cubosome. The structure here is opted by due to the this critical packaging parameter, which is almost near about 1, which is more in case of these liposomes or neosomes or herbosomes, we can say. So the cubosomes are the most stable because they have the less surface free energy and provides a huge area for the drug loading.
Well, it would be good if later on you also see the bioavailability of the potential drugs which are already in the stage of being converted to oral. And diabetes is a very good one in mind. Insulin, okay, fine, you have to inject it. There are different types of insulin and things. There are many anti-diabetic which are already in the market. But then there are also, for example, what we have seen is, you must have heard about these oral anticoagulant drugs, right? Rivaroxaban, edoxaban, which people can take. Now, the problem with them is, of course, they have a very long period of what acting in the body. We cannot have anything against them, but it would be good to test how the bioavailability for these medications would change with your cubosome preparations. Okay, thank you.
A very nice presentation, madam. Recently, we are seeing several advancements in nano formulation of these kinds. Recently, there is a nano flower that's being used for wound healing. So I have one technical question. There are several interfaces, I guess, water to polar to non-polar interfaces, I guess, for this one. So after drug loading, the drug is generally the phytocompound which you're loading. After loading and after the release, are there any changes in the structurality of the phytocompound? Has that been checked?
Yes. This structure may burst after release of the drug.
No, I'm talking about the structure of the phytocompound, which is that this nanosphere is okay. Nano cube is okay.
Ma'am, he's asking that did you check the compatibility of all the ingredients with each other? That is
We
the
have
FTR
studied the compatibility study. I have shown also. We have like we have done DSC. We have done
No, DSC is for the stability of the compound. That's fine. But the structure, is it changing the structure because of the loading in the interfaces and all those? It's not changing the structure. And is there any changes in the type of bacteria that are found on the skin, the natural bacteria? Has that been checked?
No, not yet.
Very nice work, madam. Thank you.
It's just an initial study. We are.
I understand. I saw your papers. It's good. It's promising. So wonderful work. Thank you.
Thank you, ma'am, for your wonderful presentation. I also have a question to you.
Okay.
Actually, I want to ask you that what is the basic differences between this cubosome and the phytosomes? I think I was very confusing that you use the phytochemicals. So I think the term would be better. It is say phytosomal cubosome.
Yes. Very good question. Actually, it first came into our mind that why this cubical structure when there is already these liposomes or nanosomes or herbosomes are there. They are having spherical structures, and we all know that spherical structures are the having minimum surface area or like that. But cubosomes are more stable because I have just said to sir that there is a critical packaging parameter of every compound, and this depends upon the structure of the lipid. The lipid which we are using here, GMO, it is the ratio. Critical packaging parameter is the ratio between the lipid polar part of the lipid and the non-polar part of the lipid. When it is near to 1, it forms a cubical structure by itself in contact with when it comes in contact with water.
Okay. Is the surface morphology was checked through the transmission electron microscope? Or
no. We have sent it, but the reports I don't have yet. Yes.
In this case, the results are abated still.
Pardon?
Morphology results are abated, na?
Morphology, I have SEM, but I don't have TEM.
Okay. Thank you. Thank you so much.
Thank
you.
Yes. Ma'am, I have one question. What are the key advantages of cubosomes over the nanoparticles drug delivery system, ma'am?
Yes, the first and foremost advantage is that we can upload both the hydrophilic as well as hydrophobic drugs, because there are it's a bicontinuous aqueous channel within the cubosomic structure. So this is the foremost. Then we can, since the particle size is very low, nano size, we can it can penetrate easily. We can use it for topical formulations and made up of GMO, that is glyceryl monooleate. It is a plant derived lipid, so it is biocompatible. All these are.
Okay, thank you, ma'am. Hello, very nice presentation, Disha. Disha is my little sister.
Thank you, sir.
So, if we take a tablet formulation or capsule formulation, after dissolution penetration through the mucosal barrier, it enters into the bloodstream. Through bloodstream, it enters into the target tissue. And then afterward, that drug metabolism started. But for nanosome, cubosomes, they are the drug structure or formulation structures, in nano level. Definitely, their structure molecular size is far lesser than that of the tablet or capsule oral formulations or injectables. So does it that for metabolic during the drug metabolism for nanosomes, cubosomes, body has to pay less energy? You got my point?
Yes.
What is your opinion? I don't know. This is I am in curiosity. That's why I am asking.
Sir, I think so, but I have not gone through some paper like that. But I think yes, the body.
This may be also added advantage for this formulation, sir. That is very expert.
The theory behind one you do for nano particles particularly, when basically it is all depends on the surface area increases.
Yes.
And this theory is not only for a drug, for everything. So you have HPLC,
Yes.
So what is there? Why you have size? Because more is absorbed. The surface area increases. In that way, because now you have suppose you have a sphere and then you make several spheres, small, the total surface area will increase. And once total surface area increase, then wherever it is going, there is a kind of an interaction, absorption. So that increases, means it gives more surface. That is the basic philosophy of coming to smaller and smaller particles. The more smaller particles, the surface area increases several times. And that is the reason why in simple, I'm saying chromatography. What is HPLC? We say high pressure. It is basically this particle sizes become very small. So the compound which you want to, purify, it is absorbed, the starry surface area. So efficiency increases everything. So that is the whole philosophy. The same thing is going on in our body also.
Yeah.
So that will be the reason. Should be the reason.
Thank you, sir.
A nice presentation.
Thanks.
I'm just curious. This question may not be directly to you, but anybody to answer. Once a drug is developed in lab, it is found effective. What steps you take, or how much time it takes typically to come to the marketplace so that can be used?
It's a long.
All talks, it used to be about 15 to 16 years.
Yeah.
But nowadays it has come down to say 11, 12 or 10 years. So not less than 10 years. Very rare it can be. So this is a very long process. And the preclinical part means that when you are designing a drug, making a drug, synthesis and passing vitro models and vivo models, vivo means animal models, then toxicity. It's a very lengthy process. Toxicity means if suppose you have to give a drug for more than a month, suppose like diabetes drug, then you have to do 6 months toxicity. If the drugs are which have to be given for a week or say 2 weeks, then 1 month toxicity can work or 3 months. So this toxicity data, all this goes, it is a preclinical part. And once it is selected or it is being presented, just like IND committee member, we were for 10 years. Yesterday, YK Gupta was there, Rath was there. We are all experts on the national level. And then this data is presented. And once this data is okay, then we give permission, the IND committee gives permission or expert committee gives permission for a phase 1 study, which I made on human beings. So it is basically about 5 to 6 years preclinical part if it has been done efficiently. Otherwise, it may take many of drugs they pass. But in phase 1 to phase 2 and phase 3, this is a longer time, far longer. So it takes up almost 8 years. Because you have to get the patients, first of all, human volunteers healthy is okay, but then you have to have the patients and then efficacy, then different centers. It's a big protocol for all.
Thank you, sir, for your insight and guidance. Thank you very much.
Thank you, Disha. Thank you very much for your nice presentation. Swati ma'am, please.
Thank you, Disha ma'am, and thank you to the chairpersons. Now, I would like to request that honorable chairpersons, Professor Dr. Mridula Saxena ma'am and Professor Dr. Shishir Nandi sir, to present a memento to Disha, speaker of the session, Dr. Disha Datta ma'am. And I would like to request Rajan sir to show the e-memento of Kuljit Chaudhary ma'am and Dr. Chumule. Now, I would like to request Professor Dr. Deepak Tewatia sir and Dr. Aruni sir to present a memento to the chairpersons of the session. Thank you, sir. Thank you, ma'am. Now, we have organized a short tea break. So I request the dignitaries and participants to have a tea, and we'll join the conference hall at after 10 minutes.
So now it is the tea break. So I request all the dignitaries to please enjoy the teas and come back within 10 minutes to the seminar hall. And we would like to enjoy the online oral presentations of the young researchers. The most energetic session will be now. So please come back after 10 minutes.
Hello, 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 hello. All the participants are requested to join as a panelist so that they would be able to present their session. I once again requested to all the online presenters to accept our request as a panelist. Hello. Hello. All the presenters are requested to keep ready your presentation as we have promoted to the panelist panel. So I once again request you to please be ready with your presentation.
Hello. Hello. The tea is ready, so I request the dignitaries and students to please join us for the tea. Hello.
I once again requested all the online presenters to be ready with your presentation. And as I have provided you the control panel, so you should open your PPT in PowerPoint presentation and then go to the slideshow. And there is an option browse by an individual, and then start the slideshow from the current slide, and you will be able to share through the Zoom in a proper manner. So these are the instructions for all of you, and we'll be ready after 15 minutes. So be ready with your presentation.
Welcome everyone. So we are going to start our online oral presentation session, for which I would like to request and I would like to invite Professor Dr. Deepak Tivatia sir and Dr. Disha Datta ma'am to chair the session. Professor Dr. Deepak Tivatia sir is the Director of Global Institute of Pharmaceutical Education and Research, Kashipur, and he's also the chairman of the Symposium, and he has many research and review articles in his account. And Professor Dr. Disha Datta ma'am is as a professor in the Devasili Vidyapeeth. And over to you, sir.
ये सब ओरल प्रेजेंटेशन होंगे ऑनलाइन है
सर, यहाँ तक ओरल प्रेजेंटेशन है 11 तक।
अच्छा
और ये दोनों का ये पोस्टर होगा लास्ट में। ये इवैल्यूएशन शीट है सर। ये दोनों का ये जो आस्था सायनी उनकी है, ये आस्था यहाँ पे लिखेंगे और सायनी यहाँ पे। बाकी हो गया है?
पेन दे दीजिए।
ये सर
इन दोनों का क्या है?
ये दीजिए जरा
ये पोस्टर है मैडम, ये देखो तो तुम तुम्हारा इसकी इवैल्यूएशन यहाँ लिखना, आस्था सायनी का यहाँ पे।
सबसे पहले ये अक्षय कुमार है ना, सबसे पहले ये है अभिषेक कुमार मिश्रा है।
पहले
ये बाद में है?
ये बाद में है।
अच्छा तो ये जो है
कल नहीं हो पाया था, वो आज होगा।
ये पूनम गंगवार है ना सबसे पहले, इनके बारे में बोलना चाहिए
ना, कुछ बोलना नहीं।
कितना टाइम
आपको सब
मैडम है तो सही, मैडम करती रहेंगे क्वेरी।
सर टाइम कितना देना? टाइम लिमिटेशन होगी ना?
हाँ
टाइम क्या दिया जाए? ये 1.5 घंटे का सेशन चले, 1.5 घंटा।
और उसके बाद वैलिडेशन भी तो होगा, उसके बाद लंच होगा। लंच होगा ना 1:30 का है, 1:30 का है ना, तो आप 1:30 लगवा दो लंच। हाँ अगर 10-15 मिनट हाँ वैलिडेशन होगा तो 1:30 बजे लगवाओ। सुनो 1:30 बजे अगर लग जाएगा तो कोई बात नहीं, हम लोग 15 मिनट लेट भी पहुंचेंगे तो क्या फायदा। 1:30 बजे लग जाना। हाँ लंच लगता है तो लंच तो फिर चलता ही रहता है।
हाँ सर, मैं इंडिकेट करूंगा, मैंने आप भी देख लीजिएगा एक बार।
नहीं हम मोबाइल पे कर लेंगे। 10 मिनट
8+ मिनट प्रेजेंटेशन होगा, 2 मिनट उनकी इंट्रोडक्शन। ठीक है
स्टार्ट करें सर। शुरू करें?
जी सर।
Good morning to all of you. So I am very happy to say that now we are in our last technical session, oral presentation by the young researchers. So in this line, I would like to call upon the first presenter, Poonam Gangwar. Poonam Gangwar is not present. So Aparna Joshi.
Hello, am I audible?
Hello, good morning, madam.
Good morning, sir. Am I audible?
Yes, you are audible. And now you can start your presentation, please.
Okay, sir. Yeah. A very good morning to all the dignitaries and the chairperson of the session. My name is Aparna Joshi and I'm a research scholar at SGR University, Dehradun. Yeah. Today I'm going to present a small part of my research work, that is a comparative analysis of agenidipine co-crystal formulation by two different methods, that is solvent evaporation method and wet grinding method. So first of all, I would like to discuss the objective of my study. The co-crystals of agenidipine tablet were formulated using two different techniques, solvent evaporation and wet grinding method. I have already added a picture of prepared co-crystal. Maybe it's not that clear or visible by online, but it's very clear when we see the co-crystal with the naked eyes. And after formation of co-crystal, I have confirmed the formation of co-crystal with infrared spectroscopy. After confirmation of formation of co-crystal, there is a comparative study. We have done a comparative solubility study for both the for the prepared co-crystal by both the methods. Now I would like to introduce my drug. I have taken azanidipine as an ideal drug for the formulations. Azanidipine is a dihydropyridine-based calcium channel blocker. It is used to treat ischemic heart disease and cardiac remodeling after myocardial infarction. But azanidipine suffer with a low solubility profile. That's why I have choose the drug azanidipine. And nowadays, co-crystals and co-crystallization is a recognized method to enhance the solubility of any poorly soluble drug. That's why I have choose the co-crystal formulation. So in this study, the poor solubility of azanidipine has been addressed to formulate co-crystals using saccharin as a co-former, because in the formation of co-crystal, a co-former is a necessary thing. I have used saccharin as a co-former, and the formulation were prepared in 3 molar ratios, that is 1:1, 1:2, and 1:3, using 2 different methods, that is solubility evaporation and wet grinding methods. Now, I have prepared co-crystals. First of all, I will discuss about the solvent evaporation method. The formulations, as I told, the formulations were prepared in 3 different ratios, that is 1:1, 1:2, and 1:3, where the 1 is the amount of drug, which is constant, and I have varied the amount of co-former, that is saccharin. In this study, I have used 8 mg of azanidipine because the recommended dose of azanidipine is 8 to 16 mg once in a day. And I have used 8 mg because in the further research studies, I have already I'm going to compare the marketed formulation of azanidipine, which is available in 8 mg, and I have used the solvent standard solvent methanol because my drug is soluble in methanol. Same, I have prepared the formulation with the wet grinding method in same ratios using same solvent. Again, after preparation, first of all, I have checked the co-crystal with the naked eyes, and then I have confirmed the formation of co-crystal with the IR spectroscopy, which can be seen with this picture. In IR spectroscopy, different peak shows, like in 3680 to 3650, OS stretching, CS stretching, carbon-carbon stretching, which confirm the formation of, and I have also seen the peaks for co-crystals, the saccharin, and drug azanidipine. The different peaks confirm the formation of co-crystals. After confirmation by IR spectroscopy, further physiochemical studies were performed, which were the major study for melting point. We can see in the table the individual melting point of azanidipine, that is 193 degrees centigrade, and the individual melting point and the melting point of for saccharin is 228. And we can also see that in the next table, where the co-crystals formed by both different methods, azanidipine and saccharin co-crystal crystal melting point. We can see here clearly by the graphs or by the table that there is a depression in the melting point of the formulation of azanidipine saccharin co-crystal, which also confirms that there is the formation of new crystalline form. After that, we have done the solubility study for all prepared co-crystals in different media and different pH. With the graph or with the table, we can see that there is a significant. First of all, I have performed the solubility profile in water, and in the further table and the graph, we can see that there is a significant difference in solubility by both the methods, but the highest solubility increment have been shown in solvent evaporation method in phosphate buffer 7.4. So we can say that both the method by wet grinding method and solvent evaporation method increases the solubility of azelnidipine, but the highest solubility has been seen in azelnidipine saccharin 1:3 in phosphate buffer 7.4. At last, I would like to conclude my presentation. That is, as I have discussed earlier, with the IR studies, with all the peaks indicate the chemical interaction between the saccharin and co-crystal, which confirms the formation of co-crystals. And there is a depression of melting point reveals multi-component system and designated formation of co-crystal. And saturated solubility studies show the significant increase of solubility of drug by both the methods, and there is a significant increment in the solvent evaporation method. Thank you very much for the patient listening, and now I would like to answer if there is any question.
Thank you very much, Aparna.
Thank you,
It's a
sir.
very nice presentation. Now I would like to say that if anybody in the audience have any query about the topic. Has anybody got any query? No. Aparna, I want to ask a little bit of inquiry that you told us now that you get the maximum solubility in phosphate buffer.
Yes, sir.
So what mechanism do you feel that why the solubility is good in phosphate buffer?
Sir, I have used the solubility in different, as I told, in different pH and in different solution by flask shaking method. So as I have checked, then I have seen the maximum solubility in pH 7.5. So I don't know the mechanism. I don't tell exact what is the mechanism, how the phosphate buffer, maybe the pH, maybe the solvent show the highest solubility.
Aparna, nice presentation.
Thank you, ma'am.
And I will have some queries. Like, I want to know whether your drug is having polymorphic forms or not. Means I don't know. I'm just asking.
No, ma'am, no.
Okay, only one crystalline form it is present. Okay,
Yeah.
so you are using saccharin to form this co-crystals. So what about the side effects of saccharin if we consume it orally or co-crystals?
Yeah, ma'am. I have done a lot of research because in many research, I have seen that saccharin at a specific dose show carcinogenic effects.
Yes,
Like,
yes, that's why I'm asking.
Yeah, because in case of saccharin, it's usually used as a sweetener also. Like we are saying, like we are using a sugar-free thing, which is we are using saccharin in sugar-free tablets also. But I have lots of research paper, and it says when we take like 500 mg dose of saccharin once in a day or for 1 month or 1.5 month on daily basis, then it's not a carcinogenic. It's a carcinogenic at a specific maximum, like 5 mg when we take once in a day, it's carcinogenic. But lower than 5 mg, it's not carcinogenic. That's why maybe the manufacturer is also using this.
Means
Yeah.
why you have selected saccharin? Is there any specific reason or in the
No, ma'am.
literature you have found?
Actually, I have also used two other co-crystal co-formers, ascorbic acid and salicylic acids, and I have done this study with 3 co-former, and in all of 3 co-former, I choose saccharin the best, because saccharin showed best results with the with other studies like DSC, SEM. I have also performed the other studies. So in all of the three, saccharin showed best results with azanidipine. That's why I have done this presentation, or a small part of my research is with this. I have also
thank you.
data for other, when I will publish a paper, or I will present the other part of my research, then I will also add two other co-format. But my best results I got with saccharin.
Okay. Thank you.
Thank you, ma'am.
Okay. One more question, please.
My question is, why do you say saccharin sugar-free?
Yeah, sir, I have not, maybe I have said that, but I have seen in many of the sugar-free tablets, or in many of the sugar-free juices, in many of the sugar-free products.
That's my question. Why it is? Yes, you are correct. You have said
Yeah.
or not, but people say those who take diabetes fellows, they take saccharin. I mean, say sugar-free.
Yeah.
Why do you say sugar-free? While it is also, it stays sugar.
Yeah, it stays sugar.
Sweetener.
Yeah.
So why? What can be the reason for this?
I don't know the exact answer for that, sir.
What normally the sugars, what do they contain? What is they are made up of? Sugars, you have glucose, sucrose.
Yeah, glucose, sucrose,
They are all
lactose.
sugars.
Yeah,
So
yeah.
what they are? They are the property. What are they? In terms of chemical names broadly, just if some say alkaloids, you say something other. So what do you say for these sugars? Even I would say in some extent, your chapati which you are taking from wheat, that can also be sugar.
Yes, sir, I think in case of saccharide, there is no carbohydrate. Lots of carbohydrates in the sugar.
Yeah, but the reason is all these sugars, they have carbohydrate. The carbohydrate saccharin, the structure is not carbohydrate. Do you know the saccharin structure?
Yes,
Chemical
sir.
structure of saccharin.
Yeah.
What does it contain? It is a phenyl ring associated with NH and SO, and so there is no sugar. In sugars, carbohydrates, carbohydrate structure, general structure issue. That is the reason. My other question is that you say buffer. Now, buffer is there, but which pH of buffer? What was the pH of the buffer?
7.4.
Yeah. Why do we take 7.4?
Sir, to create environment which is inside our body.
So it is, you should say it's a physiological pH. So
yeah.
Okay.
Hello, Aparna, nice to see you after a long time.
Thank you, sir. Same here. Nice to hear you.
You are brightening. Thank you very much for your nice presentation.
Thank you, sir. Thanks a lot
ha-
for inviting me.
What is the side effect of saccharin? Long-term use of side effect, does it cause any problem, health problem?
Sir, I have seen that when it's taken once in a day, maximum to maximum the 5 mg per daily dose, it cause carcinogenicity, or it cause liver damage.
Okay.
Yeah.
So, do you suggest the use of saccharin in drugs and food additives?
Sir, I can suggest, because I'm using saccharin in my formulation, but we have to check, we have to control the doses in the food products and the other things in which we are using saccharin. It's not harmful in controlled doses, but when we take at higher doses, it's obviously harmful.
Does it, sir? Sir? Saccharin has been reported as renal impairment
sir.
and
Yeah.
this liver toxicity also, hepatotoxicity.
Yes, sir.
Even
Yes,
in
sir.
small doses also it may be harmful. It has already been reported.
I have seen some articles,
Yeah.
but I have
So-
seen that after 5 mg of daily
Would
dose.
it would be suitable to incorporate it as a, your formulations like that?
Hi, Aparna. Thank you for the presentation.
Thank you,
I think I
sir.
can help you from that perspective.
Thank
The
you,
main
sir.
thing is when we see, in fact, as a clinical pharmacologist, we see the cumulative dose accumulation. So like, for example, your paracetamol is a liver toxic drug. It is
Yes, sir.
commonly used for poisoning in Europe. Every day we see. So if you exceed, if you take your paracetamol tablet, which you get in the market about 50 to 60, then we have to see the plasma levels of it, and it can cause permanent liver damage. I think the saccharin, once, if there's any cumulative dose, which is when you're taking it regularly, that would be considered in that. But as far as normal saccharin use, beyond, within the limited, especially if you don't have a liver impairment, suppose somebody has a liver cirrhosis, on top of it, he is taking saccharin as a sugar-free, then certainly it would be more harmful for him because liver is already weak, the capacity is weak. Or if it's somebody is taking with a bad kidney function, ibuprofen, for example, regularly, for them, ibuprofen is a poison. But for a person with a healthy kidney, it might be not, if taken at regular intervals. So I think that perspective you might have to add when you do your next presentation, that what is the cumulative dose, which is known for this drug to show side effects. Yeah.
Okay, thank you, sir.
Okay,
Thank you for your suggestions.
Our next participant is Archana Dhyani. Archana Dhyani, can you hear me? Is she connected? Hello, Archana. Okay. Hello, Archana. Are you listening?
Yes, ma'am. Good morning, ma'am. Am I audible?
Yes, you are audible. Please share your presentation.
Okay, okay, ma'am. Let us stop.
So your time start now. You have 10 minutes.
Just a second, ma'am. इससे मत कर स्क्रीन शेयर करनी है. Hello, good morning, ma'am.
Good morning. You start.
Where is the PPT? Just a second, ma'am. Just a second. I'm facing some problem. Just a second. Yes,
Okay,
ma'am.
your presentation is visible. Now you start.
Okay. Okay, ma'am.
You
Fine.
can start.
Good morning, everyone. My name is Archana Dhyani, and my topic of presentation is formulation and evaluation of drug phospholipid complex for solubility enhancement. The presentation is divided into the different parts like introductions, materials and methods, results, and conclusion. Now, moving towards the introduction part, first I would like to introduce what is drug phospholipid complex. So, the drug phospholipid complex is usually defined as a complex in which the drug molecule will get entrapped inside the phospholipid moiety, and the interaction between the drug and phospholipid, it will be results into a complex formation. These drug phospholipid complex, they will offer the various benefits over other lipoidic drug delivery systems like liposomes, because in liposomes there will be a chances of the drug leakage, but in drug phospholipid complex, there will be a presence of some bond formation between the drug and phospholipids, which will be remains so that the drug will be remains in the complex. And these complexes are like biodegradable, non-toxic, safe, That's why they are also preferred. And the third basic advantage of these phospholipids is that they will be shows the same absorption mechanism as that of triglycerides and phospholipids in the body. Despite of their various advantage, they will also offer certain disadvantage, like these phospholipid complex is highly sticky, and which will be results in the clump formation, as well as which will results in the non-flowing properties. So here the phospholipid complex, the solubility of phospholipid complex or the stickiness can be avoided by converting into or by incorporated into a matrix carrier, which is consists of an polyvinyl alcohol. Here my drug, which is used for the studies, clinidipine, and the phospholipid, which is used for the studies, eglecithin. Clinidipine, it comes under the BCS classification 2 drugs, and its pharmacological uses includes as antihypertensive drugs, and it comes under the calcium channel blockers category. Phospholipid here uses an eglecithin. So the different formulation of drug and phospholipid was prepared, and the ratio varies from 1:1, 1:0.5 to 1:1.5, and they are subjected to a different temperature, that is 40, 50, and 60 degrees Celsius. And similarly, the stirring should be carried out for different time intervals, that is 3, 4, and 5 hours. The method which is used for formation of the phospholipids complex is solvent evaporation method. So what will happen in solvent evaporation method? The drug and the eglecithin was mixed in RBF in the different ratio, and then they are stored at 300 degrees Celsius, 300 RPM at different temperature and for the different time intervals. And once they have formed, they are subjected to rotary evaporator so that the solvent can be evaporated, and then the phospholipid formed is dried in a desiccator. After that, they are incorporated into a polymer matrix, that is of PVA, which will be helps in to remove the stickiness of the polymer. This slide shows the preparation of clinidipine phospholipid complex. Now moving towards the results part, when the drug phospholipid complex matrix has been formed, they are subjected to different evaluation parameters like percentage yield, entrapment efficiency, drug polymer interaction, particle size, and zeta potential, and in vitro release studies. So first I have to talk about the percentage yield and entrapment efficiency. When the percentage yield was calculated, it was found that the maximum percentage yield was found for the formation F1, and similarly, when the entrapment efficiency was done, it was found that the maximum entrapment efficiency was found for the F1 formulation. And we can say that the formulation F1, which will consist of a drug on phospholipid ratio 1:1, it shows the good percentage yield as well as entrapment efficiency, where the temperature was done at was put at 50 degrees Celsius, and the time of stirring is 4 hours. The next evaluation parameter is FTIR study, which was done to find out the interaction between the drug and the complex formation. The first FTIR, it will represent the clinidine phospholipid complex. And the FTIR shows that the characteristic peaks of the clinidipine, it is all present into the complex, and additional peak was been found in the complex that will be shows due to the bonding between the drug and the phospholipid. And the second figure, which will shows the FTIR of the drug, FTIR of the polymer, FTIR of the matrix dispersion. And from this FTIR, it was found that all the peaks which is present into the complex, which is also retained here, and there was no disturbances of peaks has been observed, which will shows that the drug and phospholipid complex do not interact with the PVA solution. The next evaluation parameter is particle size. The particle size of clinidipine phospholipid complex and matrix dispersion both was determined, and from the result, it was found that the particle size of the formulation clinidipine phospholipid complex is 261 nanometer, which is having a PGI of 0.492, and that of matrix is particle size of matrix is 329 nanometer, that is having a PGI of 0.549. Then the results of zeta potential shows that the range of zeta potential in clinidipine phospholipid complex is minus 0.3.36 to minus 6.6 and having an average value of minus 0.4.46. And in matrix dispersion, it is found to be between minus 19.4 to minus 20.4 and an average value of minus 19.9. And this shows that the formed clinidipine phospholipid complex and matrix dispersions both are stable. The next result is in vitro drug release study. The basic rationale of this work is to enhance the solubility of the drug clinidipine because it comes under the category of the BCS class 2 drug. So the in vitro drug release study was done, and from the figure we can see that there are the three graphs, which first one is of drug, second one is phospholipid complex, and third one is matrix dispersion. So from the graph, it can be clearly understand that when the matrix dispersion was formed, it is having a higher dissolution rate as compared to drug and phospholipid complex. So initially the drug will shows a poor solubility. After that, phospholipid complex, it will shows a better solubility, and when it is incorporated inside the matrix dispersion, it will shows a more solubility, which will results to a better dissolution, and it will ultimately leads to enhancing the oral viability of the drug, because the oral viability of drug is low because of it falls under BCS class 2 category. So when the data it is put into the various mathematical models, it was found that the most of the formulation, it will follows a supercase transport 2 mechanism, that is due to the swelling and the relaxation of the phospholipids. At the last, it was concluded that the clinidipine phospholipid complex and matrix dispersions can be easily prepared by solvent evaporation method, and the solvent evaporation method shows the enhancing the aqueous solubility of clinidipine by 2.5 folds when it is incorporated inside the matrix dispersion. Hence we can say that the drug phospholipid matrix dispersion complex, it can be used to enhance the solubility of a poorly soluble drug. Thank you.
I just have one question.
Yes, ma'am.
What about the stability of this your complex, phospholipid complex? Have you gone for any stability study? Because in these type of complexes, I have seen a stability problem, and for this I have seen this cholesterol. I have gone through some papers. I have seen cholesterol are using in order to make the formulation stable. So what is your planning about for this study, stability studies, or have you gone through this release profile also?
Yes, ma'am. The stability study and the in vivo study, it will be my future plans, and it will be done after some period of time.
What about the release profile? You have not explained
release
that.
Ma'am,
Which
release
profile it is following? Zero order or first order? Which model it is following?
Ma'am,
That
it
you
is
also
following
have
zero
provided.
order kinetics. Zero order.
Zero order kinetics.
Yes, ma'am.
And what about your zeta potential you have said?
Yes, ma'am. Zeta potential, both matrix dispersion will having a separate value, and phospholipid.
What is it
will
the matrix system value is?
It's minus 19.9.
Okay,
It
and what
is stable.
Okay, yes. What is the range for the zeta
Range is
potential?
between minus 19.4 to minus 20.4.
No, I just want to know what is the range for zeta potential? Do you know for stability
Yes,
of
ma'am.
colloidal dispersion?
Yes, ma'am. Yes, ma'am. Ma'am, it's like is range between 19 to 20. It is stable, and above that, the value will be get enhanced. That at this time I don't recognize or don't remember the exact value. But this
Okay.
range, at this range, the formulation
And
is
what
stable.
about your polymer phospholipid complex? Phospholipid and drug complex,
Okay.
zeta potential?
Okay. Yes, ma'am. It is having a value of minus 4.46. So it is also stable category. It is more stable than matrix system.
Okay. Sir,
Archana, is a very nice presentation by you.
Thank you so much, sir.
Archana, I want to ask just a very simple question. Is there any specific reason for choosing this particular drug that you have chosen for looking into the enhancement of the solubility by
Sir,
this
because
solubility? Have you gone
sir,
through any
this
literature that this drug is having some problems in bioavailability? Previously, it is available.
yes, sir. Yes, sir. So I have gone through the literature review, and this drug will having a oral solubility of almost 13%. That is due to its solubility problem. So that's why I have particularly choose this.
Okay.
one more question, sir.
Yes, ma'am.
Yes. Archana, first of all, very good presentation it was. And I have a question to you. Can you show me the conclusion slide, please, on the...
Okay. Sure, sir. Sir, is this visible?
Yes, yes, yes.
Yes, sir.
Right. Actually, you said, I think clinidipine is a BCS class 2 drug. I think
Yes.
I am right now. Okay.
Yes, sir.
So,
Yes, sir.
so what will be the application of this complex? Actually, this drug is already available in the form of the tablet in market. So why do you formulated this complex, and what are the application in future for this complex?
So, although the drug is available into the market, but it is
Yes.
having a poor viability problem. So, my major concern is to enhance the viability of the formulation by making it into a complex formulation.
Okay, but as far as I am aware about the BCS class 2 drugs, they are highly
Yes, sir.
permeable and they have the low solubility. So to improve the
Yes, sir.
solubility, you formulated this complex. And
Yes,
how
sir. Yes, sir.
how it will be delivered into the body?
So, orally, by an oral route.
Okay, thank you so much. Right.
Okay, sir.
Okay, thank you, Archana. Next one is Asha
Thank you so much, ma'am.
Gumadi. Next participant is Asha
Yeah.
Gumadi.
Yeah. Am I audible, ma'am?
Yes, yes. You can share your PPT.
Yeah, yes, ma'am. Sure. Is my screen visible, ma'am? Hello.
Yes, yes. You continue.
Yeah. Morning, everyone. Myself, Asha Kumadi. I'm a research scholar in KVSR Siddhartha College of Pharmacy, Andhra Pradesh. Today, my topic I'm going to explain about is comparison of 2D and 3D cancer cell cultures. What are the different types of 3D cells and its characteristics? So why this is going to be my future PhD work? Why I've taken this 2D and 3D cell cultures because nowadays most of the regulatory bodies banning the in vivo testing. Like in June 2024, European Pharmacopoeia, they banned the rabbit pyrogen test to prevent the animal work. So what is the next alternative method is cell line cultures. For the, it could be research purpose or drug discovery process. So before going to my topic, first I'll briefly explain about what is the difference between 2D cell culture and 3D cell cultures. 2D cell cultures is nothing but we are the regularly what we grow the cell lines are the 2D cell cultures where we are going to grow our cells, required cells, what it could be primary cells or any other cancer cells, we are going to grow in the cell culture. It could be, they may be of adherent type cells or cells suspension culture cells. When it comes to the 3D cell cultures, there are of two different types. There are scaffold-free system and scaffold-based system. Here, the scaffold-free system includes spheroid techniques, organoid techniques, whereas in scaffold-based systems, solid scaffolds and hydrogels are there. Here, these spheroids are nothing but the tumor-like cell clusters that helps in developing the natural gradients of the oxygen and nutrients. So they form an aggregation, formation of an aggregation cells and together form a tissue-like structure. And organoids also a different type of 3D cell cultures. Here, these are nothing but a mini tumors. These are derived from the different stem cells that are closely resembles the actual tissues which are present in our body. When it comes to the scaffold-based system also, there are two types are there, that is solid scaffolds and hydrogels. These are prepared by taking some scaffold, some matrix materials that could be of collagen or any other based protein materials. In between, these cells are going to be grown and form a solid scaffold. In hydrogels also, they're forming a complex structures. So the main mostly difference here are the cell interactions. That is here in 3D cell cultures, there are extensive mimicking the in vivo conditions. That means they are like a same like a in vivo. We are maintaining they're having both in vivo conditions, and here we can also determine the resistance of our drugs towards the real tumor cells also in the 3D cell cultures. Whereas they are the genes which are expressing, which are mostly similar to the actual tumors which are present in our body. And 3D culture is also very expensive compared to the 2D cell culture. So first we have to know what is the EMC is. EMC is a extracellular matrix environment that helpful in supporting the cells and growth in a tissue form. We can see that in the previous slides also, they are like tissues, bunch of cells are complex to and form the tissue-like structure. So this extracellular matrix that helpful, like which includes proteins, glycoproteins or polysaccharides, which provides a structural support and biochemical support also. Like how it is going to support means they are helpful in the formation of the 3D cell cultures by taking a different, by using different materials like collagen, fibronectin for creating the natural tumor microenvironment. And this is also helpful in the regulation of both cells, adhesions, migration, and the proliferation and differentiation. And we can also check the progression of a cancer also. And they also helpful in the nutrition and oxygen distribution, which creates the same similar what the conditions which are present in our body. And these are also helpful in can act as a barrier to drug penetration. And from this we can check whether there is any similarity happen in the real tumor also. We can know that. The examples here are the hydrogen scaffolds and the bioprinted tumor models. How it exactly useful in the cancer research? First, it is, as I've already discussed earlier, that these are creating a tumor microenvironment outside the body. And we can determine the, because they are resembling the tumor, and we can work on the cancer models very easily. And they helpful in improving the drug tests and resistance studies. It is when we use a 2D cell culture, it is somewhat difficult to detect or predict the drug response, whether it is showing any resistance is there or not, how exactly the drug is going to work or not. It could be somewhat difficult in 2D cell structure, whereas this one is going to helpful in how it is going to be stimulating the drug penetrating barrier, and how it is showing the resistance. All these things we can know, and we can also understand the what is the chemoresistant mechanism which could lead to the better treatment strategies. And this also helpful in the personalized cancer therapy. How this is going to helpful is we are going to take the cells of or cells or tumor tissues from the patient, and we can grow the patient derived tumors, and we can check whether if we give some particular anti-cancer drug, and we'll check what is how it is exactly going to work on the particular patient tumors. And we can check with this, what we can do is we can better approach treatment process can be different to the based upon the patient condition. And it is also helpful in studying the tumor progression and metastasis. Like models like a bioprinted tumor tissues or hydrogel based cultures, they help us to know how the cancer is going to spread based upon different type of cancer and different persons. Like we can know that if I taken up cells from the patient, and I can check how much the cancer has been spread or not, and based upon that we can give the dosing process or anti-metastatic drugs can be given to that particular patient. And this main purpose is helpful in the reducing the animal testing, because nowadays it is ethically not correct, and so hence ethical alternative method, best method is this 3D cell culture. And this is also helpful in the immunotherapy research. The best example for the 3D cell culture where it is being helpful is 3D cell culture. That is, we are going to co-culturing. Co-culturing means culturing two different cell lines. That, like here, what we are culturing is culturing of cancer cells with the immune cells. With this, what we can know that whether how our
Asha, wrap up fast. Okay, your time is going.
yes, ma'am. So here I'm going to check whether how the immune cells are being interacting with the cancer cells. With this, we can improve the therapeutic efficacy and improving the treatment strategies. So here what are the till now real life applications which have 3D cells are there. Here the spheroids like obtained from the oral cancers showed that a proportion of cancer initiating cells probably increase in the proportion of cancer initiating cells. It could be because of the epithelial mesenchymal transition process that is occurs in 3D cell cultures. And some of the 3D scale scaffold systems are here. One example is that it is showing that resistance of paclitaxels in the 3D cell cultures, not only in the 3D, not only in the cancer, they're also helpful in identify the pathways. For example, here the primary human salivary gland cells are there, where we can determine the secretion of saliva or any other protein secretions. We can, for that purpose also, we can use the 3D cell cultures. And here one more method is co-culturing system, which I've already discussed in before. That here we are going to co-culture with two different types of cells. From this, we can distinguish between what is my target cell and what are the assistant cells are there, which are supporting the growth and the development of that target cells. And it could be of co-culturing, could be of different of two types. It could be direct or indirect. From this, we can determine, and we can also grow the two different cultures by keeping a physical barrier. With this also, we can determine whether physical barrier can...
your time is finished. So
yeah.
You just conclude in 1 minute. Okay.
Okay, ma'am. From this, I conclude that 3D cell cultures provides a more physiological relevant models which could be closely mimicking the tumor cells, and which could be valuable for the advanced cancer research and personalized medicine. These are my reference. Thank you.
Thank you, Asha. So my question is, which model is better according to you, and why?
Here, it could be different, depends upon the cancer model which I'm working on, madam. My suggestion is we can go for the spheroid based type cultures, because they are resembling the same actual tissues of the tumors, which could be helpful in the identifying the better understanding of the tumor progression.
Asha, very nice presentation by you.
Thank you, sir.
I have a little bit of confusion that what is the basic difference between 2D and 3D culture media for developing the cancer cells, culturing the cancer cells?
Yeah, it could be depends upon the type of models which are creating, sir. Here, 3D models normally media with any 5% or 10% serums will be there. Whereas here in 3D cell culture also, we are providing the media and the need whatever the things which are required. But here we are adding some other materials. For example, if the scaffolds are there, we are going to add collagen materials, or we are going to add glycoproteins or some polysaccharide materials for its supports of its growth to form a 3D structure. Yeah, but the medias will be
This is
same.
about the media, what you are telling about. I am asking that what basic thing makes it two-dimensional and three-dimensional?
Here, in 2D cell models, normally we are growing the cells, whether it could be adherent or suspension. But whereas in 3D cell culture, what happens is, along with the cells and the media, we are going to add the aggregate materials. For example, low jellying agarose for the formation of a round-like structure to create this 3D structure. Or if we add the collagen of different concentrations, we may form different other scaffold-based signals.
Okay, thank you. Any questions from
Yeah.
audience?
Hello. Hi, I am Dr. Saxena. I'm one of the oncologists sitting in the room. I
Yes.
just wanted one suggestion in one sense. You are using 2D and 3D cells. You are basically growing the cancer cells in vitro
Yes.
conditions, right? And
Yes. Yes, sir.
trying to stimulate its behavior similar to a real cancer cells which you have in a patient. Now,
Yes, sir.
first of all, how I think the availability of cancer cells from biopsy samples of the patient should not be a challenge. So you would not prefer using the real cells in vivo medium to do the studies? Why we need 2D and 3D structures when we have plenty of cancer cells around us?
Yeah, we can work. But if you have biopsy materials with me, but most of the researchers like me who want to work on, so for the further research also, this 3D cells will be very useful, sir. Like every time biopsy results will not be there, like us. So for that, these techniques are going to be helpful for the research.
Okay, is it like in sense of there's a lot of resource requirement to culture
Yes.
real cells
Yeah.
in the
Yeah.
medium, or a cost component? The reason why I'm asking is, and I think it would be good for the audience too. You must have heard like cancer coming back, cancer in metastasis, cancer not responding to treatment. And we have seen patients, I see particularly breast cancer, lung cancer, and the possible mechanism, even with the immunotherapy, which is a very promising word, that immunotherapy is very great and good. There are many side effects associated with it. The peculiar thing about cancer is the cancer is the only cell in the living world, very similar to virus, which has its own intelligence.
Yes.
That's why a cancer cell can be getting can be responsive to chemotherapy. When I give first cycle, second cycle, third cycle, suddenly there's no response. That means cancer starts growing in spite of getting chemotherapy as recommended. And the reason is the mechanisms which the cancer cells can develop to fight against that chemotherapy are at different levels. Some cancer cells develop channels we call as efflux channels in their cell membrane. Just the medication goes in, it pumps it out. Some cancer cells modify their cell wall and cell structures during the phase of chemotherapy itself, that they become non-responsive to treatment.
Yeah.
And for us as clinicians, it is always difficult to explain to patients why suddenly I'm not seeing any response to a patient when I'm giving treatment. First 3 cycles, excellent response. Then suddenly cancer grows, even though the patient was on chemotherapy. So what I mean to say is, of course, I appreciate and I know that your researchers or many researchers are using 2D, 3D simulated cancer models for developing the things. But the main problem which you would face and why the things would fail is because your 2D, 3D structures doesn't have intelligence. And either you change it, as in real world it happens, because I see so many. And then we say, if you involve an oncologist, we say, okay, if somebody even was responsive to a treatment, and then suddenly after 7 months he comes again and we find it is metastatic, we still try to do the same chemotherapies. That's why you have different chemotherapy lines, like first line, second line, third line. In breast cancer, there are 18 lines. And we expect for one line to be effective for 6 months. So we say a lady who is in a good age of 40 that potentially you might be able to live 18 cycles we have, each cycle holds the cancer for 6 months, that would be a potential lifespan. Of course, previously we never had many so. But what I mean is that you need to really think about that you're doing your so much of efforts in 2D and 3D structures was absolutely great. But this potential challenge you would feel when you would come in vivo because
Yeah.
cancer cells are intelligent, simple.
Yes, sir. Thank you for the suggestion, sir.
I mean, this is a very important points which we see when it comes to practical implications. Yeah.
Yeah, I understood, sir.
So-
I'll try to include this in my work so that I could check. Can we first check whether whether how would drug is acting continuously? I'll look into it, sir. Whether I'll check whether I can create any new model to determine or to check whether how this process is going to work, why it is not working in the further chemotherapies. Thank you. Thank you for the suggestion, sir. I'll work into it also. I'll include in my work process.
Thank you very much, Asha.
Thank you, sir.
I think you might be getting some good points and knowledge about your work.
Yes,
and
sir.
really going to help you. I am very much
Yeah.
thankful to Dr. Aruni also that he is here with us and
Yeah.
he has imparted such a great information and the
Yeah.
basic knowledge about the cancer.
Yeah.
So our next presenter is Geet Gupta. So Geet Gupta is online.
Sir, am I audible?
Yes, you are audible. You can continue, please. Share your presentation, please.
Yes, sir.
Yes, now your screen is visible. You can start. Your time starts now.
Good morning, sir, and chairpersons. Myself, Geet Gupta. I'm doing masters from Applied Science, Applied Chemistry in MIT University, Lucknow campus. My presentation is for the topic synthesis, characterization, and antimicrobial screening of novel hydrocyclic 2-oxo-2H chromine, 3-carbonyl, spiroindolin, and 3,3-pyrazolidine 2,1 base derivatives. My abstract is on that I have made and synthesized and screened a series of novel pyrazole spiro-based cyclized derivatives of acetyl, iminoethyl, and ethiniol. All these compounds were screened for antimicrobial activities. The final derivatives were procured by initiating the condensation of precursor molecules, salicylic and ethyl acetoacetate, from acetyl coumarine. Then the products were allowed to react with substituted astatine, which then yielded chalcone. And if we do the further reaction of chalcone with hydrazine hydrate, aminoguanidine, HCl, and thiosamicarbazide, then we got final products as 1-acetyl 5 substituted, 2-oxo-2H chromine 3-carbonyl, and many more. My introduction would be I've done with spirocyclic compounds and bioactivities. Spirocyclic compounds are prominent bicyclic compounds in which two cycles are joined together by a common atom. The organic spiro compounds are a significant class of compounds found in naturally occurring stuffs by extremely prominent bioactivities. These compounds have also exhibited additional properties like antimicrobial activity, antifungal activity, and antiviral activity, and anti-cancer activities, and many more. Many natural and biologically active compounds and drug molecules contains organic spiro compounds as one of the most important constituent. And then I'll talk about 4-amino-1,2,4-triazole, which I've used. It's one of the most important hydrocyclic compounds due to their pharmaceutical and biological activity, like anti-tubercular, anti-tumor, and anti-leishmanal, anti-convulsant, anti-cancer, and anti-inflammatory, antimicrobial, and antimicrobial activity. Small ring hydrocycles constitute scaffolds in medicinal chemistry. For generating biologically active derivatives, data indicate that at least 85% of bioactive compounds contain a high heterocyclic moiety. Then I'll talk about steroidal spirocyclic compounds. Steroidal spiroheterocyclic compounds are promising drugs for diverse disease varying from hedgehog signaling inhibition for the treatment of cancers and prevention of tumorigenesis. Synthesis of spiroheterocyclic steroids is facilitated by stereochemistry of steroids for generating single diastereomers. Pyrazole moiety. The pyrazole moiety is a five-membered heterocyclic structure with nitrogen atoms is widely used. Pyrazole is a five-member heterocyclic compounds containing two adjacent N atoms. It possesses biological activity including pesticidal, herbicidal, and anti-inflammatory activity. My research extends the synthesis of novel pyrazole spiro-based derivatives including carboxyamide, carboxymetamide, and carboxythiamide derivatives. I've done the synthesis by using 20 mmol OL salicylaldehyde and ethyl acetoacetate with absolute alcohol diethylamine with reflux with continuous stirring for 4 to 5 hours, and the product was filtrated and then washed with excess water in dried air and recrystallized from ethanol 3-acetyl. Then I synthesized 3-acetocoumarine. The yield which I got is 85% and its melting point is 120 to 121 degrees Celsius. After that, I also and the compound which I made right now is 3-acetylcoumarine is denoted as 2AC in this slide. And by 2AC, I have synthesized three more products, which is 3AC, 4AC, and 5AC. I'll start by 3AC. For the synthesis of compound 3AC, which is 1-acetyl, 5-substitutase, 5-2-oxo-2H chromine, 3-carbonyl, spiroindolene, 3-pyrazolidine, 2-1. For that, I have used 0.01 ml of 2AC compound and 20 ml 1.4 dioxane and hydrazine, hydrate in presence of acetic acid as catalyst, reflexed on heating mantle for 8 to 10 hours. The reaction of mixture was cooled, poured into crushed ice, used HCl for the neutralization. Then I got my third 3AC compound, which was filtered, dried, and recrystallized from ethanol. Further, I've also done this by I've also synthesized compound 4AC, which is 5-substituted 1-1 iminoethyl, 5-2-oxo-2H chromine, 3-carbonyl, spiroindolene, 3-pyrazolidine, and 2-1. For this, I have taken 0.01 mole of 2AC and 0.01 mole aminoguanidine, HCl 20 and HCl 20 ml, dioxane, which was refluxed on heating mantle for 12 to 13 hours for the reaction mixture was cooled, poured into crushed ice, and used acid HCl for the neutralization. Then the PPT, the 4AC, I synthesized 4AC, which is dried and recrystallized from methanol. So as such, I have also synthesized the compound 5AC, which is 1-ethanol, 5-2-oxo-2H chromine, 3-carbonyl, spiroindolene, 3-pyrazolidine by 2AC. For this, I've taken mixture of 2AC, 0.01 ml and thiosamicarbazide with thioxane and refluxed heating metal for 7 to 8 hours, and then reaction mixture was cooled, poured by HCl, dried HCl, acid HCl, and then filtered and recrystallized from ethanol. In this table, we can see that, and we can see vitro antimicrobial activity for newly synthesized pyrazole spiro-based derivatives of carboxyamide, which is 3AC, carboxymetamide 4AC, and carbothiamide 5AC against positive gram and negative gram fungus. Yeah, and my result and discussions. My result would be that I've synthesized synthesis of novel pyrazole. I've done the synthesis of novel pyrazole spiro compounds, synthesis of 3-acetylocoumarine, formation of chalcones, and synthesis of pyrazole specific compounds. My spectral analysis would be that I've done IR, HNMR, and CNMR. IR vibrational bands for NH2 I've seen at 3118 to 3500 centimeter inverse. For C double bond N in 1560 to 1589 centimeter inverse. and for S double bond C in 1318 to 1370 centimeter inverse. I've done HNMR for CH2 protons in 2.98 to 3.30 ppm and confirming pyrazole rings in 2.98 to 3.30 ppm and CH protons in 5.28 to 6.00 ppm and CNMR signal at 40.33 and to 45.54 ppm and CH2 for CH2 in 154.85 to 178.58 ppm. These are my references which I've used for writing the paper. And thank you so much. And you can ask me the questions.
Thank you very much, Geet Gupta. A very nice presentation. Now I would like to open for the questions from the audience. Any questions? So Geet, I want to ask that you have only done the antimicrobial activity on fungus only.
Yes, sir. I've done it. I have not done it. I'm a chemist. I'm a chemistry student. I'm not a biologist. So my co-colleague have you done that? I've synthesized the compounds.
Okay.
Which strain of fungus you have used and what culture media you have used?
I'm sorry. I don't know about it. I'll read about it further.
Okay, okay. Okay, nice presentation, Geet. Thank you so much.
Thank you.
Next one is Hina Faruqi. Is she there? Hina Faruqi? She is not there. Next one is Neetu Rajput.
Good afternoon, ma'am. Am I audible?
Yes, you are audible. Please share your presentation.
Yes, ma'am.
Share it fast.
Yes, ma'am. Just a minute.
You have limited time.
Is my screen visible, ma'am?
No, it's not.
Just a minute, ma'am. Is my screen visible, ma'am, now?
No, it's not.
There's some network issue actually, ma'am.
So what you can say it verbally. You just explain it without the slide or.
Okay, ma'am. Just a minute. Now it is sharing, ma'am. Is it visible now, ma'am?
Yes, your mobile screen is visible. Make it
Okay,
fast.
thank you, ma'am. I'm Niti Rajput, research scholar at Graphic Presentation is formulation and evaluation of methyl methacrylate grafted copolymer of neem gum by grafting technique. So my main the research work mainly focused over the modification of the natural polymer as nowadays natural polymer is gaining more interest in the field of pharmacy, but they also suffer some disadvantages related to their flexibility, compatibility, degradability, and rigidity, solubility, etc. So in order to enhance these properties, modification of natural polymer is required. So number of methods are available for the modification of polymer, out of which I have selected the grafting method for the modification. of natural polymer, as grafting involves the attachment of a monomer at the polymer backbone in order to enhance the properties of the main natural polymer with the use of microwave radiation and the free radical inducer. So I have selected neem gum as the natural polymer and methyl methacrylate as the monomer with the use of ferric ammonium nitrate as a free radical inducer by using microwave assisted grafting technique. So here is the formula for the formulation. And I have varied the concentration of the free radical inducer as well as the monomer, and the microwave time is constant. So after the formulation, I have evaluated the grafted copolymer through various techniques like percentage grafting, percentage efficiency, water retention capacity, hydration capacity, and also the FTIR and DSC analysis. So the result which I concluded was the grafting has been occurred and the polymer properties has also been modified in terms of the water retention capacity, hydration capacity, swelling index, viscosity, so that it can be used in the formulation of further dosage form, whether it is a sustained release, control release formulation or nanoparticles. And also the grafting occurred up to a stage as the after addition more of the monomer and the free radical inducer, a saturation at the polymer backbone has occurred, due to which the grafting is terminated. So the batch 7 showed the best grafting, which is going to be used in the formulation of further doses form. And my research work is also going on further. So my conclusion was this. Thank you.
Nice presentation, very fast going. Okay, I have a query. So, have you extracted this neem gum?
No, ma'am, I have not extracted. I have purchased from the market, as my main focus is on the modification of the natural polymer. So neem gum already being published as a extracted and already being tested. So I have already purchased.
Okay. So
Yes,
which
ma'am.
technique you have used for grafting?
Microwave assisted grafting technique, ma'am.
Okay, and what is the future formulation you have? Means what is the application of making this?
Ma'am, I'm going to use this polymer as the neem gum has wide application as a thickening agent, as a binder in the formulation of sustained release, control release. So my plan is to formulate a sustained release tablet, or also I'm planning to formulate a nanoparticle also, which is I'm going to plan. Actually, not decided.
Okay, okay. Okay, nice presentation. Over to you, sir.
Thank you.
Okay, any query from the audience? Okay, thank you. Next participant is it's Nirvesh Chaudhary. Nirvesh Chaudhary. Yes, Nirvesh Chaudhary.
Yes, ma'am.
Share your presentation.
Okay, ma'am.
Yes, we are.
Good morning, chairpersons. I am Nevesh Chaudhary, PhD Research Scholar of TMU Muradabad, College of Pharmacy. Today, I am presenting my topic on a review progressive lipid-based nanocarriers to treat the neurological disorder. So the introduction is the recently developed novel nanotechniques have been employed for the sustainable treatments of various diseases like neurological disorder, including Alzheimer's, Parkinson's, schizophrenia, depressions, and epilepsy. In neurological disorders, the existence of the blood-brain barriers functions to safeguard the central nervous system from the varieties of toxins. Most antipsychotic drugs create adverse effects which conventional drug delivery to antipsychotics therapies. New medicines-based treatments targeting the specific brain area involves in CNS biology. Novel formulations is required to improve the treatment of strategies and support the research and developments of the novel ones. New nanoformulations like solid lipid nanoparticles and nanostructured lipid carriers use to minimize the problems of antipsychotics drug released to a specific site at the lower concentrations. These lipid carriers show a great potentials for great release of the drug through the brain by using the specific route. The next is methodology. The developments of the lipid-based nanocarriers involves the applications of spray drying, solvent emulsification, solvent evaporations, ultrasonications, and homogenization technique. The best techniques is homogenizations followed by ultrasonications. The nano lipid carriers are prepared by melting the lipid phase above 100 degrees Celsius, at melting temperatures of solid lipids and mixing continuously strings with aqueous phase containing the surfactant, followed by the ultrasonications and homogenization techniques. These techniques provide the nanoparticles in the aqueous phase less than 200 nanometers is suitable for all types of delivery through the different route. And the result and discussions, the nanolipids carriers have emerged as the promising drug delivery systems and provide their unique properties. Nanotechnologies have facilitated and developed the nanostructures lipid carriers for other routes, such as the intranasal drug delivery system, offering benefits like improving drug stability, targetings, and penetrations. Research and development efforts may be focuses on the NLC systems with enhance the loading capacity and improving stability due to their compatibility with the BBB layer. The nano-size lipid carriers systems is the ideal solution for the medications delivery to the central nervous system. The easy incorporations of drug molecules into the neuronal cells results in the enhanced stability and great concentrations. And the conclusion is, this approach improves therapeutics outcomes by enabling safe and effective targetings in chronic neurological disease treatments. In this studies, we delve into the current advancements in nanotechnology and neurological therapies and explains their mechanisms of actions, the NLC features, and uneven internal structures of the lipid systems that offer better drug incorporation. It is essential for enhancing the viability and permeation rates of the lipophilic molecules and increase the drug solubility with lipid components and minimize the side effect of the lipid dosage forms. So, thank you.
Thank you, Nirvesh. Is there any question from the audience? Any questions? Okay, thank you. Next, we will take the next participant, Nitin Srivastava. Okay, sir. Nitin Srivastava. He is there? Nitin Srivastava, share your presentation. You will have 6 minutes.
Okay, ma'am. Very good. Very good
Within
afternoon
6
to all of
minutes, you
you.
have to wrap up, okay?
Okay, ma'am. Okay. Is my presentation visible?
No, it's not.
Okay. Just a minute. Is it visible now?
Yes, visible. You continue, please.
Okay. Okay. Thank you. First of all, good afternoon to all of you present over here. And I would like to thank GPER for giving me this opportunity to present my work. I will be speaking about the greener synthesis of quinoline using ionic liquids. Well, heterocycles are organic compounds which acquire significant position in development of human society. And most of the therapeutics that mimic natural products are belonging to the heterocyclic classes. And many advanced action against diseases and their diagnosis is achieved by designing and testing some structures which are basically heterocycles. And in such case, heterocycles play very important role. Various biologically potent glycosides, alkaloids, antibiotics, pesticides all belong to heterocycles. Among these heterocycles, quinoline ring is a part of a natural product, and it is pharmacologically very active molecule and used in many drugs. These diverse classes of drugs are indispensable part of human life, and quinoline moiety is found in anti-tubercular, anti-malarial, anti-cancer, anti-HIV, antiviral, anti-inflammatory, analgesic, antihypertensive, anti-Alzheimer, antibiotic, anti-leishmanial, antioxidant, antiprotozoal, DNA, like this. There are so many drugs which belong to this category have quinoline rings. And these are some of the drugs. You see the structure. Most of the drugs here like quinine, quinidine, chloroquine, mefloquine, sparfloxacin, ciprofloxacin, irinotecan, camptothecin, vasnaricone, saquinir, bedaquine, divocaine. Divocaine is a local anesthesia, primaquine, topotecan, which is anti-cancer. These all drugs we see here that they contain quinoline ring. That is why quinoline synthesis is very important in the pharmaceutics. So quinoline is basically benzopyridine or azanaphthalene, and it contains some quinoline hydrogens, which contains C double bond NH2 moiety. It exhibits polar and non-polar properties which are helpful in transmission through the bacterial cells. These are some of the approaches which have already been done for the synthesis of quinolines. These are Conard, Limpach, Dobner, intermolecular cyclization, radical-induced rearrangement, Kiesinger synthesis, Poirot synthesis, Friedlander synthesis. There are so many types of synthesis which have been carried for the synthesis of quinolines and substituted quinolines. So we talk about the UN SDG, which are Sustainable Development Goal, and these are given by United Nations in 2015. And these talk about 16 SDGs, which talk about the sustainable development all around the world. And in this SDGs, if we focus upon the SDG 9 and SDG 13, they both tell us about the sustainable synthesis, and sustainable synthesis can be done with the help of many reagents, and beside using metal catalyst or
Nitin,
organic
minute is left. Please wrap up fast.
Okay, ma'am. So in this regard, ionic liquids play a very significant role because ionic liquids are non-volatile. They possess polarity. They are green, known for green organic synthesis. They are non-volatility, non-inflammatory, thermal stability, and controlled miscibility. Can enhance the product with a greener for the greener synthesis of these quinolines. There are some of the nanoparticles, some composite, which have been incorporated for which are basically ionic liquids, and they play a significant role in the synthesis of quinolines. These are basically Friedlander synthesis, Dobner synthesis, Stroop synthesis. All these types of synthesis have been carried out with the help of these ionic liquids. So in this regard, we can say that ionic liquids play a very significant role in the synthesis of biologically potent moieties. And we can, because these are tailor-made, we can design them according to need of the miscibility. We can design them according to need of catalyzing the reaction. So in this way, we find that these ionic liquids are very important. And in case of quinoline, we find that all types of significant classes of synthesis like Stroop, Friedlander, Dobner, Bronsted acid, and multi-component synthesis can be done with the help of these ionic liquids. These are some references. And thank you.
Thank you very much, Nitin. Very nice presentation.
Sir.
Now I will call upon our next presenter, that is Ruchita Day.
Okay, thank you, sir. Thank you, sir. Thank you all.
Thank you. Hello. Yes, your screen is visible. You have 6 minutes to present your
Okay,
presentation,
sir.
please.
Very good afternoon to all the respected delegates present. I am Rishita Dey, working under the supervision of Dr. Ashmita Shamadar, Assistant Professor, Department of Zoology, University of Kalyani, and under the co-supervision of Dr. Shishin Nondi, Professor and Head, Department of Pharmaceutical Chemistry, Global Institute of Pharmaceutical Education and Research. The title of my presentation is Docking-Based Screening of Potent Natural Components to Combat COVID-19 Associated Brain Fog. So what is this brain fog? So previously we used to say that it is a term of feeling of confusion, forgetfulness, or mentally impaired, or we accept it as a part of getting older. So, but brain fog is not really a medical condition that can be diagnosed, but rather it is a warning signal of a different underlying health problem that should be identified and addressed. So, coming to the background of the study, the COVID-19 has rapidly escalated into a global pandemic, and the highly contagious SARS-CoV-2 strain that causes the coronavirus disease was found to be associated with deadly respiratory complications. The symptoms like fever, persistent coughing, difficulty breathing, sore throat, loss of smell or taste, runny nose are some of the symptoms. But although COVID-19 is particularly a respiratory disease, but it has the capacity to damage other organs, including the brain. So, this brain fog related issue has come into forefront after the outbreak of the second and third wave of COVID-19 infection. So, this COVID-19 patient exhibited spectrum of neurological symptoms indicating that it can affect the brain in a variety of manners. So, what is the route that the virus reaches the central nervous system is either through inhalation or through lung infection followed by hematogenous spread or through nasal mucosa and olfactory nerve fibers. So these are some of the combinatorial issues that appears as a result of the brain fog, like unstable mental state, difficulty in focusing, feeling disoriented, headache, and some of the neurological aspects or symptoms like ischemic attack, anosmia, and agiosa. So coming to the multiple factors that postulates or contributes to the neurological manifestation of long COVID symptom is that the persistent systemic inflammation leads to cytokine production, immune system activation, and production of reactive oxygen species. And the increased blood-brain barrier permeability also allows these cytokines to penetrate the brain and to induce the neuroinflammation. And the more porous the blood-brain barrier also have the ability for direct viral invasion of the brain, and tissue hypoxia may also occur as a result of this microclot formation. So there is no permanent solution in this regard, and there are concerning facts about the synthetic drug that is piracetam and oxiracetam. Though these drugs are very popular and have a role in brain neuronal regeneration and brain function, but in several European countries, the FDA has not authorized their approval in patients. So therefore, the focus has been shifted to an alternative solution, that is the inclusion of phytodrug in therapeutic management of brain fog, because as they have limited cytotoxicity and they are easily available and cost effective. So this is the table that shows the bioactive components along with their structure that are drawn in ChemDraw software. And then coming to the result section, I've used the two protein, that is acetylcholinesterase and butyrylcholinesterase, as they play an important role in the pathogenesis of neurodegenerative diseases. And these are the best doc confirmation of the phytodrug, that is lutein, with these two proteins. This is the best doc pose of quercetin with acetylcholinesterase and butyrylcholinesterase. And these are the best doc confirmation of curcumin with the two proteins. And this is the best doc pose of resveratrol with ACHE and BCHE. And this is the interaction of piperine with acetylcholinesterase and butyrylcholinesterase. And this is the best doc pose of linalool with ACHE and BCHE, citronella, and mahanimbin. So this is the table that shows the important amino acid residues that links the different moieties of the ligand with the
Rishita, please try to wind up in
Yes,
1.5 minutes, please.
Yes. And the respective doc scores are given below. And so coming to the conclusion, that the present study showed that luteolin, quercetin, curcumin, resveratrol, piperine, linalool, etc. have a strong affinity to bind to the brain acetylcholine and as well as butyrylcholine esterase receptors. And the common amino acid residues are phenylalanine 329, tyrosine 332, alanine 328 that interacted with BCAG, whereas histidine 447, tyrosine 337, phenylalanine 338, serine 203, tryptophan 86, and tyrosine 341 that interacted with ACAG. And this predict a common mode of binding for the selected phytoactive components to combat COVID-19 associated brain fog. And reports confirm that the low-grade and chronic inflammation is persisting in both the body and neuron of the patient, the consequences of which trail to experience of brain fog in patient. Although there is a lag in the knowledge regarding the etiology and specific treatment, however, researchers are in search of pharmacological intervention that might improve the cognitive function by targeting neuroinflammation. And in order to understand the neurological function for further improving and opening the treatment gateway, these Ayurvedic herbal medications might come as a solution in treating the problem. So I highly acknowledge University of Kalyani and Global Institute of Pharmaceutical Education and Research for providing me the necessary facilities for carry out this work. And thank you so much.
Thank you very much, Rishita. As usual, very nice work and nice presentation by you. Thank you very much. Now I will like to call upon our next presenter, that is Priyanka.
Yes, sir. Hello?
Priyanka, you are audible. You can share your screen
Yeah,
and
yeah,
you
I'm
can start your
sharing my
presentation,
screen.
please.
Yes, sir, sure.
Yes, now your screen is available, there. So you have
Yes.
6 to give your presentation.
Sir, is my screen being shared?
Your screen is visible. Please continue.
Okay. A very good afternoon to each and everyone present. So, I'm Priyanka Mazire from Department of Biotechnology, Savitribai Phule Pune University, and I'll be speaking about unmasking Leishmania donovani MAP kinase 12, a comprehensive study implies its probable role as a drug target. To begin with, I would like to give a brief introduction about the disease itself. So, visceral leishmaniasis, also known as Salazar, is still prevalent in many countries and is the most severe form of leishmaniasis, which is caused by the protozoan parasite Leishmania donovani and Leishmania infantum. So, according to WHO 2023 report, more than 95% of real cases are majorly from 10 countries, India being one of them. Furthermore, 80% of the global burden of EL is reported from the South Asian countries, particularly eastern states of India such as Bihar, Jharkhand, Uttar Pradesh, and West Bengal. The therapeutic arsenal available for the systemic treatment of visceral leishmaniasis constitutes of amphotericin B, miltefosine, pentavalent antimonial, and taramomycin. However, the available antivisceral drugs have restricted report here due to the several shortcomings, including the safety, efficacy variance, cost, and the resistance. Therefore, identifying the targets which are essential for parasite survival is imperative for developing newer potent drugs. Talking about these drug targets, Leishmania mitogen activated protein kinases have been studied extensively as a drug target. About the 15 Leishmania MAP kinases which are being reported, Leishmania MAP kinase 6, 8, 11, 12, and 15 have not been yet characterized or their function is not been reported. we can see that among the 15 Leishmania MAP kinases, the TXY motif is highly conserved in all the Leishmania species. So we will focus on the objective of this study was to determine the structural and functional characteristics of Leishmania MAP kinase 12 by the in silico study. Further, to evaluate the expression of MAP kinase 12 in high passage and low passage Leishmania promastigotes, followed by amastigote. And lastly, to see the effect of pro-inflammatory and anti-inflammatory stimuli on the expression of LD-MAPK12. So based on the in silico and in vitro studies, the results which we got are, first we will focus on the in silico analysis. The in silico analysis was carried out with the different species of Leishmania and the Leishmania donovani MAP kinase 12. It was found that the sequence is highly conserved among the Leishmania species, and the subdomains of Leishmania MAP kinase is conserved in Leishmania MAP kinase 12 of L. donovani also. The phylogram depicts that L. donovani MAP kinase 12 is highly similar to L. infantum MAP kinase 12 and L. major MAP kinase 12, while it is divergent with the Homo sapiens MAP kinase 12, which makes it as a potent drug target. Further, the secondary structure analysis of LD-MAPK12 by pre-fired server showed that the LD-MAPK12 has a higher ratio of alpha helices and beta sheets, followed by domain and binding site identification using Interpro database. It showed that the LD-MAPK12 has phenylthreonine protein kinase at domain, whereas it belongs to the protein kinase family, having ATP binding region.
Priyanka, please try to conclude in 1 minute.
Yes, sir. The in vitro study was carried out using the low passage and high passage promastigotes. So, the low passage promastigotes are known to be virulent ones, while the high passage are the virulent strains. Well, we saw that the LD expression was high in the low passage promastigotes, while the expression of LD-MAPK12 was lower in the low passage, high passage promastigotes. Followed by, we also carried out the metacyclic and procyclic expression of LD-MAPK12 in metacyclic and procyclic promastigotes. So, metacyclic are the promastigotes which are highly infectious, and we found out that the expression of LD-MAPK12 is high in the metacyclic promastigotes. We also checked the expression in the macrophages as well as dendritic cells. Similar results were seen where we found out that the expression, relative fold expression was higher in the macrophages which were infected with the low passage promastigotes.
Priyanka, your time is running out. Your time is running out.
So, last year, sir. The LD-MAPK12 expression was checked upon the stimulation of pro-inflammatory and anti-inflammatory cytokines, which showed that the expression of LD-MAPK12 reduced after the pro-inflammatory cytokines. Thus, this concludes that LD-MAPK12 is an virulence factor
Thank you,
virulence
Priyanka.
factor. Yes, ma'am.
Thank you, Priyanka. Your time is over. We will take
Yeah,
the next poster
sure.
presentation.
Yeah.
Astha for poster presentation. Astha?
Yes, ma'am. Am I audible?
Please share your poster.
Yes.
Only for 3 minutes. Okay?
Okay, ma'am.
Poster is there.
Oral presentation. Good afternoon, respected dignitaries. Today we are going to present our e-poster on pathogenesis mode of treatment, and to explore current targets of trypanosomiasis. Trypanosomiasis is a parasitic disease caused by Trypanosoma brucei and Trypanosoma cruzi, responsible for human African trypanosomiasis and American trypanosomiasis, also known as Chagas disease, respectively. It is world's 17th most neglected tropical disease declared by WHO. However, there are no synthetic drugs available to treat the disease, and this calls for prime exploration of disease pathogenesis and development of potent specific drugs targeting the parasite.
Now coming to the life cycle. Trypanosoma cruzi has two hosts, mammalian host and triatomine bug. Trypanosoma brucei also has two hosts, mammalian host and ciciply. Metacyclic trypomastigote is the infective stage for the mammalian host. Trypomastigote is the infective stage for the insect host.
To address this issue, here are some proteins that can serve as drug targets for Chagas disease. First, TCAEK1. It is present in epimastigote, amastigote, and trypomastigote form. Inhibition of this protein leads to impaired cytokinesis, higher DNA content, polyploidy, and nuclear degradation. Second, cruzipain, a major cysteine protease of Trypanosoma cruzi that protect the pathogen against host immune system. Third, FeSOD. These are iron-containing metalloenzymes that protect the pathogen against exogenous superoxide radical and provide resistance against apoptosis. Other such proteins are TCPKC and TC1.
The most important protein targets of human African trypanosomiasis are first, TBCATB, a major cysteine protease which targets on brain and liver. It obtains iron from the host body and also helps in catabolism of transferrin. Second, TB-catin, another major cysteine protease which also targets on brain by crossing the blood-brain barrier. Third, cytidine triphosphate synthetase. This protein helps in de novo synthesis of CTP and provides necessary amino groups. The other such important proteins are TR and IC75.
Some chemotherapeutics to combat Chagas disease. First, nifurtimox. Nifurtimox generates free radical that binds to T-cruzi DNA, causing damage. Others are benznidazole and fexinidazole.
Chemotherapeutics to combat HAT are
Please
first
conclude in 1
mainly
minute.
targets on IC75 and inhibit the function of DNA replication. Second, pentamidine. It targets on mitochondria and inhibit the function of enzyme topoisomerase. The other such important drugs are melarsoprol, pafurimidine, and acoziprol.
Therefore, we can conclude that there is no such broad spectrum drug available to treat Trypanosoma cruzi and Trypanosoma brucei infection. So, targeting endogenous protein of both the parasite will be helpful for development of potential drug. Thank you.
Thank you, Astha. Nice presentation. Now, Sayoni. Sayoni is there? Sayoni, please share your poster.
Am I audible, ma'am?
Yes, yes, you are audible.
Yes, ma'am. I'm sharing.
Share your poster.
Yes, ma'am. I'm sharing my poster.
You have just 3 minutes.
Good morning, respected dignitaries. Today we are going to present our e-poster on evaluation of preventive efficacy of different phytocompounds to combat hyperglycemia in mice model, a comparative assessment. Diabetes. Diabetes is a chronic metabolic disorder affecting millions of people worldwide. There are many orthodox medicines available in the market which has proven efficacy to decrease blood glucose level in diabetic patients. But side effects of these drugs affects a person most. So nowadays people are more inclined towards the use of phytocompound as drug which are relatively safer and provide better action in lower dose and targets various proteins related to the pathway of diabetes.
The objectives of our study are number 1, validation of preventive efficacy of selected phytocompound against diabetes, and number 2, comparative study of chlorphylline and curcumin against allogeneic-induced diabetes in mice model.
Coming to methodology, for our study, we focused on glucose parameters, histology of tissues, DNA fragmentation assay, COMET assay, fluorometric assessment, evaluation of person cell viability, immunofluorescence study of different proteins, and biochemical analysis. Now moving to the results, both the chlorphylline and curcumin decreased the blood glucose level of allogeneic-induced diabetic mice, but chlorphylline did better. Same results were exhibited by chlorphylline pretreated mice group in DNA fragmentation assay. In COMET assay, the COMET tail of pancreatic beta cell was decreased by chlorphylline pretreated mice group compared to curcumin pretreated mice group. In case of fluorometric assessment of sperm, it was also shown that the mice group pretreated with chlorphylline showed better result. We have also checked the persons of cell viability of hepatic and pancreatic cells. Both the curcumin and chlorphylline done good results and good effects, but chlorphylline showed better result than the curcumin.
We have also done the propydium iodide staining of pancreatic beta cell and studied the expression of BCL2 and cytochrome C, which are very important apoptotic markers on pancreatic cells. In case of propydium iodide staining, intensity of propydium iodide get decreased more in chlorophyllene pretreated mice group than the curcumin pretreated mice group. In case of expression of BCL2, BCL2's expression get increased more in chlorophyllene pretreated mice group, and in case of cytochrome C, expression of cytochrome C decreased more in chlorophyllene pretreated mice group than curcumin pretreated mice group. From the histological staining of tissues, we can also conclude that both the phytocompounds chlorophyllene and curcumin restored the tissue architecture of liver, testes, and brain after the diabetes induced by alloxan. But here also chlorophyllene gave better result. From the study, we conclude that the both phytocompounds chlorophyllene and curcumin show the good results, but in comparative assessment, the chlorophyllene show better result than the curcumin. So we can also say that chlorophyllene, which is present in green vegetables, may serve as a potent preventive drug against diabetes. Thank you so much.
Thank you, Seoni. Our last participant is Vanshika Gupta. Vanshika Gupta. So we have finished here. Thank you very much.
Our session is over, please.
Thank you, sir. Thank you, ma'am. Now I would like to request Professor Dr. Anil Kumar Saxena, sir, and Dr. Aruni, sir, to present the memento to the chairpersons of the session. Symposium, and it's my pleasure to host the 3-day symposium. The lectures were amazing to listen, and the discussions in the form of question answers were very informative. So now I request the panelists of valedictory sessions to please grace our dais. I would like to invite Professor Dr. Anil Kumar Saxena sir, Professor Dr. Aruni Saxena sir, Professor Dr. Deepak Tewatia sir, and Professor Dr. Shashi Nandi sir, to please grace our dais. So now I would like to request Honorable Dr. Aruni Saxena sir to start the panel discussion.
So, thank you, first of all, and I would like to thank the faculty and the students of JIPA who had been quite enthusiastic about this in organizing the event itself, preparing posters, and also preparing some of the presentations. One of the things which I note is, of course, students who are for whom this whole event is organized, and what we expect you to have from it is one thing in your CVs when you come in your final year, whether you're a B Farm or M Farm, everybody would ask you in the interview what exposure, what were the college activities you were involved in. And at that point of time, there should be a real evidence rather than someone just writing, oh, I took part in basketball, I took part in cricket or whatsoever, but more on academic side. You should be able and proudly say that you have exposure to international conferences, whether it was business oriented or academic oriented. That is the value added in your CV. And you will realize it when you would come in B Farm, fourth year, you go to M Farm, that people would certainly ask you. And that was the reason we expect these things come into your CV. That's why you join JIPA and choose JIPA as compared to other colleges or institutions. So that is the takeaway for you. What you can tell in front of the interview committee, what did you learn in that event? That's one part. Second part for the faculty members, there are immense opportunities in research. I know people think on the financial gains, and I can tell you with my personal experience that you have immense financial gains to being an academician if you really know how to practically make a difference in any sort of arena in research. I use so many techniques I'm an oncologist, at the same time I'm programmer, at the same time I've done 2 PhDs, the second one is going on. And everything I did gives me financial gains. So it is not at all to be thought why to do research. If you really want to have more money and fame, if you know research and you are sincere about it and you devote 2 hours extra to your regular things in a week or 3 hours, you can literally have other sources of income. You should be smart. And at this institution, you are lucky to have that research is being encouraged and you have opportunities to come up with the projects, do the things, get publications, get private fundings if you know, and work with it. None of the, even the institution which I work, it is a very bureaucratic one in NHS. It is not possible that I can do many things and I am not paid extra for doing anything. Whereas for you, it's a platform where you can have incentives and things to do the things. I think that opportunity you should take into account and get your CVs improved. Your CV would improve. Because tomorrow, if you want to go abroad or do anything else, they would be amazed that you had done so many things while being also in a small city of Kashipur. So this is an opportunity which we are giving you. So that's important, and cash it. You won't get it easily. With these words, I would still thank Dr. Nandi. Special thanks to Dr. Tivatia and Sarfaraz. Rajan, thank you for coming all the way together. There had been glitches in the technical, but of course, there's always a room of improvement. And I would give my mic now to the chairman, Saxena, to give some words, and then to Dr. Tivatia.
Dear participants, students, and those on the dais, I think these 3 days almost, or say 2 complete days, have been very exhaustive, particularly in terms of listening lectures. It is very difficult, to get the people who can speak. So you had got such a big exposure. And I think the best thing would have been that each student, each student likes it. Rather, the students crave for it. Sometimes when I go to some their lectures, in Malaysia, I went one time that people are traveling 800 kilometers once they came to know that I'm coming for lecture. So I think if you listen lectures, those who have been given, Dr. Y.K. Gupta, who gave the inaugural lecture, and thereafter the lecture, they are the authorities in India, top, in so many committees. If anyone would have you, could impress him, he can get you a job, a government job, in no time. So the time is less. Dr. Aruni has also already told you. It is you have to multitasking. And if you are not multitasking, you cannot achieve. The time comes when money showers on you. You throw the money and money comes to you. It is not that you go after the money. But for that, you have to do very hard work. If you are coming here to listen the talk, you have also paid. So if you are wasting this time roaming here and they are not listening here, not attending, then what is the use? Until unless you are coming to Jippa, you are from Jippa, most of you. So you are coming to Jippa, you are paying money, you are paying fees. And if you are not attending the class, you are not taking advantage. Then in your life, you will not be getting anything. So I think it's a thing that you have been exposed in this session, in this thing. It is a very big achievement. People don't get these kind of opportunities. People don't get the people who have given lectures. If you see, they're very, very rare. To get Dr. Y.K. Gupta here on the platform offline is a very great thing. He has no time. You give telephones, he'll not attend, because it is not that he doesn't like to have it, but he's so busy. Okay, and just now I had a call, so he told me that he's also arranging it, because he came here, he found that he should also do some service to Kashipur because of closeness to me and becoming here. So on 24th of February, there is a webinar on this. I think this thing will be shared, and later on, I think they can put it also in JIPA. And you can attend that webinar, registrations. And in this, big companies are coming, big companies, big pharma companies, including drug controller. And this, he is arranging every 2 months, 3 months. And I think if we get chance, he says it will be better that your students, your faculty become part of it, and we'll find it out. I think he's doing as a social service. You have started a institution. He is also doing something, Dr. Y.K. Gupta. So I think he had sent that. So try to be there, expose, you should make your exposure. And then I think you will get things. We would like that our JIPA students rise in their areas. And my students have gone to a salaries, which I say 96 lakhs rupees per month. Some of them, not all. And some of them to 60 lakhs per month, I am telling, in India. But they had worked hard. So do that. With these words, I give you this thing that you have come and you have participated. So thank you so much. And I will also give thanks and appreciate the hard work which has been done by the staff of JIPA, the director, Dr. Tewatia, Dr. Shishi Nandi, Dr. Sarfraz, and all others, including our old Mr. Rajan, who is, of course, now it is in merit, but I think he has the affinity, and I thank you very much for this. All work has been taken, the responsibilities, learn from them. Organization of anything is it's not easy, and we have to learn. So we had big committees. I think all the committee members, he's looking after food committee. He's and all those who have seen the cultural program, and I think she has been here all the time. So everyone of JIPA, I appreciate and I thank them for all this cooperation. Thank you very much.
Good afternoon to everybody. Although if I have to speak about this international symposium, this is the second international symposium organized in the by the joint venture by Global Health Techno Management Forum and Global Institute of Pharmaceutical Education and Research. It is really a great achievement for us, and I'm sure that everybody has been exposed to such a great and informative and intellectual lectures during the past 3 days, that everybody is going to be benefited by those informations, lectures, and scientific sessions. But these things, I would like to say that it is only because of our patron of this symposium, our Honorable Chairman, Dr. Anil Kumar Saxena, whose guidance is always persuading us to do these things. If we are not getting such a, you can see the focused this thing, we will not be able to do all these things. So we are very thankful to Dr. Saxena. And same time, I would like to thank Dr. Aruni Saxena for sparing the time, and he's here with us in this conference, because this Global Health Techno Management Forum is only the brainchild of Dr. Aruni Saxena. And this Global Health Techno Management Forum, you might have seen in last 3 days, that this time in this conference, we are not only having the lectures regarding the drug design and only from the pharmacy background, but we have very good lectures from IT field. You can say the machine learning, the artificial intelligence. So all these things are being focused, and means they are giving us an idea that how the future is going to be and how the things will improve in future because of these types of implementation of the things like machine implementations and the artificial intelligence. So as a chairman of this conference and director of the institute, I would like to thank all the dignitaries, all the distinguished speakers, and our honorable chairman, sir, secretary, sir, Mr. Rajan, who has come for this conference for us, all my organizing committees, heads, and all the committee members. And the last but not the least of my students, who have really, they have done a very good job this time, and they have come to Ramnagar to attend this conference. And I am telling you, my dear students, that these type of the things will going to help you in future in your career. Because if you get the exposure of these type of events, you will first of all get the knowledge that what is being going on in the world in the particular area. Second thing, you will also get the opportunities to come to interact with such an intellectual people. As Dr. Saxena has just said, that it is impossible to make such great eminent scientists to be physically present over here. So we are very lucky for it, sir, and we are very thankful to all. From all my JIPA staff and faculty members, we are all thankful for to you. And we will look forward for such type of events in future also. And we will do our best to make it successful and memorable events. Thank you very much.
Now it's a moment to take you in my heart. Means the moment to give vote of thanks to all the dignitaries for grand success for this international symposium on global trends in health and technology management 2025, in continuation with the first international symposium on GTHTM 2024 organized at University Auditorium, Dehradun. I thank Dr. Aruni for guiding the Global Health Techno Management Forum formulated by our institute, and he is the backbone of this forum, always guiding us to give an international platform for all professionals to lead the health technology management and mankind. I thank all the session chairpersons, authors, speakers, poster presenters, oral presenters for covering all the burning areas of the research. I thank my organizing committee, Sarfaraj, Anjali, to take care of the registration desk. Sarfaraj is co-organizing secretary of the symposium. I thank the travel committee, Arun, he look after the travel, Munir and Sandha for looking after the foods, Vinita, Fija, looking after the students for organizing the beautiful cultural program to enhance the flavor of our Indian culture. And Dibbangshu, Adil, Rajababu for students' regulation of learning. And how can I give my thanks and gratitude to my younger brother Rajan to be with us. Rajan, you have been painted in our heart. Still, he is in mirror, but he is always present with us. Special thanks, Rajan. And my colleagues Sehjad, Rahul, Farid for taking care of the online platform. And I don't know how can I give my thank to Malakshmi, Sati, Rukaiya, and Vinita, Sandha for the stage management. And Arjunji and team is there for looking after the sound management without any interruption. Special thanks to you. I thank the media persons for coverage of our symposium news in different news channels. Special thanks to our Honorable Director, Sir, Dr. Deepak Teotia, to guide us in a dynamic way. Sir, special thanks to you. We are lucky, really lucky to have our God-gifted Honorable Chairman, Sir, Dr. Saxena, to shower a constant blessings, knowledge, I don't know. So, Sir is our academic Guruji, and I pray to God for his long, healthy lives. It's our opportunity to learn from Sir. And, special thanks to CSIR, HRDG, central government for funding, partial funding to make this grand success, to support us. Special thanks to Themis Medicare, Intas, Soligi, and Entamo. These are all pharmaceutical and health companies to be with us for sponsorship and membership of our forum. All presenters are eligible for submission of the full-length papers. You will receive email. Special thanks to Springer because articles will be published in the Springer based on rigorous review process as per the rules. And, we are every citizen responsible to take care of our globe, making more green and greener, to make eco-friendly and free from environmental pollution and toxicity, to fit for the health, because health is wealth. And I don't want to linger my lecture, and it is not the last one, but this is the beginning that say no goodbye. We are all connected by heart. special thanks. I don't know how to explain by words to Madhukarji, who is, one of the major guides of our forum, Health Technology Management Forum. Thank you, sir, to be with us for your constant support. And, already Director sir has told, we are all connected by heart. Without students' cooperation and participation, the event never be success. So the success goes to you. And Director sir already announced that we are ready to accept this kind of challenge. Now, I request Honorable Chairman sir and authority of the institute to think when next event will be organized soon, because this is our dream to make our program grand success with the help of the young scholars and students like you, because you are the main pillar of our family and you are the real heroes of our country. Salute to you. Salute. Thank you very much.
Thanks so much, sir. Now moving forward, we are going to facilitate the winners of Young Scholar poster and oral session. So I would like to request Chairman sir, Professor Dr. Anil Kumar Saxena sir, to announce the
Hello.
results.
So it is, the results are offline posters. The first, I think we go. The third prize is shared. We go.
Yes, okay.
The third prize is being shared by Arnab Chakravarti from Kalyani University. You are there? And Sandhya Dhakia of Jippa. So congratulations. The second prize goes to Divya Negi. It's from Amrapali University. And the first prize goes to Vinita Pandey. Congratulations.
So now I would like to request the winners of the session to please come on the stage.
Result is of online oral and poster presentation. Online posters. Oral as well as poster. So this is online session. The first prize goes to Rishita Day. They are being shared again by Shita Day and Astha Saha. Okay. And second is Manani Bhattacharya.
Manani Bhattacharya.
He is from
Kollam University.
Oxycontin. And Aparna Joshi. And the third is for Abhishek Mishra and Sayoni Dam. Where they are from?
Ioni is from Kollam University.
So all these prizes are mostly gone to Kalyani University, 5. And this Abhishek Mishra, also from there.
No.
Nobody is present over here.
No, they are all
online.
My certificate will be sent to their email.
Yeah, I think they will be sent to their emails. It's okay. So with this, I think one time again, I thank you all. And so there should be big applause for all organizing committee for this.
Now I request the dignitaries on the dais for a group photograph and I would like to request the students to come on in the front row for the group photograph. I request Bharti ma'am and I request ma'am and sir, please come on the stage for the photograph. Ma'am.
मैम आप आगे आ जाइए मैम आप
साजन को भी बुला लो। एक और ठीक है। खड़े हो जाओ भाई। Okay. Thank you.
So everybody take positions for national anthem, please. Everybody please take position for standing positions to honor our country by praying the national anthem. So let's start. Everybody, please silent, be stand. Start the song, please. Jana gana mana yodhinayako jayo he Bharat bhagya vidhata পাঞ্জাব সিন্ধু গুজরাট মারাঠা দ্রাবিড় উৎকল বঙ্গ বিন্দ হিমাচল যমুনা গঙ্গা উচ্চল জলধি তরঙ্গা তব শুভ আশিস মাগে তব শুভ আশিস মাগে গাহে নব জয় গাথা জনগণ মঙ্গলদায়ক জয় হে ভারত ভাগ্য বিধাতা জয় হে জয় হে জয় হে জয় হে, জয়। Thank you. Jai. I request all the dignitaries to please enjoy the lunch now.